Have you ever wondered what it would be like to witness the moment when life discovered it could see? When the first eye opened in Earth's ancient seas and suddenly, for the first time in 4 billion years, the universe beheld itself. Can you imagine swimming through warm Cambrian waters where every creature you encounter seems designed by an alien imagination? Where five-eyed Opabinia glides past on its strange proboscis? Where Hallucigenia walks on stilts that scientists once thought were its back? Where evolution seems drunk with possibility? What would you feel brushing against you in those primordial seas? Perhaps the tickle of trilobite antennae exploring this strange visitor, or the gentle current from Anomalocaris swimming by, its huge eyes scanning for prey, or the soft touch of sea pens swaying in ancient currents, filtering mysteries from the water. Have you ever watched a flower bloom in time-lapse photography and felt that surge of life exploding into being? Now imagine that explosion happening not with petals, but with entire body plans. Not in days, but in millions of years that passed like heartbeats in geological time. The greatest diversification of life our planet has ever seen. What would it sound like, this explosion of life? Perhaps the whisper of countless creatures burrowing for the first time. The susurrus of new kinds of swimming. The deep vibration of Earth itself quickening with possibilities never before imagined. If journeys through deep time bring you the same wonder they bring me, if you find peace in these ancient memories, please consider liking and subscribing to follow along for more. And if someone you care about might appreciate drifting through Earth's memories as they drift off to sleep, why not share this gift of wonder with them? I'd love to know where in the world you're listening from tonight and what time you're settling in for this journey. Leave a comment below. Let me know what corner of our modern Earth you call home, and if there are other moments in deep time you'd like to explore together, other ancient memories you'd like me to share. Tell me in the comments. Your suggestions guide our future travels, but for now, let's leave the modern world behind. Settle in now. Let the weight of your day become a distant memory. Breathe in the quiet of your room, and with your exhale, let the present moment gently drift away. We are not bound by clocks or calendars tonight. Together, we will step outside of time, to a place I remember well, a world waiting in the deep past. I am the Chrononaut, and I have drifted through the ages like smoke through still air. Tonight, I invite you to join me on a journey, not forward, not back, but deeper, into the memory of Earth itself. Close your eyes if you haven't already. Feel yourself becoming lighter, untethered from this moment, ready to slip between the seconds and find yourself in a time before time was counted. Come with me now. Let us cross the threshold together. We are traveling back 541 million years to a moment when life discovered possibilities it had never imagined before. This is the dawn of the Cambrian period, the beginning of what your scientists call the Cambrian explosion. Though explosion is perhaps too violent a word for what I will show you, think of it instead as a flowering, a symphony, a grand improvisation where life explores every possible variation on the theme of existence. I have come to show you the moment when nature became an artist, when evolution discovered not just survival, but creativity, when the seas filled with forms so wondrous that even now, 500 million years later, they seem like creatures from dreams. But we begin just before this explosion, in the twilight of the Ediacaran, to understand the quiet world that is about to transform. I find myself floating in the warm seas of the late Ediacaran period, 550 million years ago. The world here is peaceful, almost meditative in its simplicity. The water is crystal clear and warm, heated by a sun that seems younger, softer than the one you know. The temperature hovers around 85 degrees Fahrenheit. A perfect, endless summer that has persisted for millions of years. There are no continents as you would recognize them. The land masses are clustered mainly in the southern hemisphere, with a few scattered islands and volcanic archipelagos dotting the vast expanses of open ocean. The largest continent, which your scientists call Gondwana, stretches for thousands of miles, but even it would seem alien on your maps. No grass softens its shores, no trees shade its hills. The land is bare rock and sand, painted in shades of red and brown by iron oxides, broken only by the green-black stains of bacterial mats in wet areas. But it is in the sea that life has made its home, and what a strange home it has become. Around me drift the Ediacaran fauna, Earth's first experiment with large, multicellular life. They are unlike anything in your modern oceans, following body plans that evolution will soon abandon, as if life is still learning the rules of being large. Here is Dickinsonia, perhaps the most successful of these early giants. I watch one glide along the seafloor, its oval body rippling with a slow, hypnotic motion. It's nearly 4 feet across, but less than a quarter-inch thick. Imagine a living pancake, quilted with raised ridges that run from a central axis to its edges. It has no eyes, no mouth, no limbs, no obvious front or back. It simply is, a flat expanse of tissue that somehow coordinates itself to move, to grow, to reproduce. I follow this Dickinsonia as it grazes across a meadow of microbial mat. The mat is a complex community of bacteria and archaea, layered like a living carpet. The top layer is green with photosynthetic cyanobacteria, harvesting the sunlight that filters through the clear water. Below are purple sulfur bacteria, then chemosynthetic species, each occupying its preferred chemical niche. Together they form a miniature ecosystem, complete in itself. Dickinsonia feeds by simply lying on top of this mat and absorbing nutrients through its ventral surface. I can see the outline of its previous feeding spots. Oval depressions in the mat, where the microbes have been digested away. It's a slow, peaceful form of grazing, with no violence, no chase, no escape. Dickinsonia moves perhaps a foot per day, leaving a trail of grazed patches like footprints in a bacterial meadow. The water around me is filled with other Ediacaran creatures, each stranger than the last. There floats Charnia, named after Charnwood Forest, where its fossils were first found in your time. It looks like a fern or a sea pen, with a frond-like body up to 6 feet tall swaying in the gentle current. But Charnia is not a plant. It cannot photosynthesize, nor is it a modern coral or sea pen. It is something else entirely, a form of life that found a way to be large without being complex. I examine a Charnia colony more closely. Each frond is composed of branching units that repeat in a fractal pattern. Branches upon branches upon branches, each a scaled version of the whole. It's anchored to the sea floor by a holdfast that looks like roots, but serves only for attachment. The frond itself is the organism, filtering something from the water, nutrients, dissolved organic matter, microscopic prey. Even watching it feed, I cannot be certain. Swimming past is something that might be an ancestor of jellyfish, though it follows no modern body plan. It's circular, with a threefold radial symmetry that gives it an alien appearance. Tribrachidium, as your scientists call it, pulses through the water with an odd, rotating motion, its 3 arms creating spiral currents that may help it feed. Or perhaps the currents serve some other purpose entirely. The Ediacaran fauna keep their secrets well. On a rocky outcrop, I discover a garden of Swartpuntia, creatures that look like soft-bodied sea anemones, but are probably unrelated to any modern group. Each is about 6 inches tall, with a cylindrical body topped by what might be feeding tentacles, or might be something else entirely. They don't sting. Nothing in this era has evolved the complex cells needed for that defense. They simply sway with the current, dozens of them in close proximity, creating a forest of gentle, waving forms. The seafloor itself is covered in trails and burrows, evidence of creatures I cannot see directly. Some trails are simple grooves, perhaps made by something like a worm pushing through the sediment. Others are more complex, paired grooves suggesting multiple legs or appendages, though what creature made them remains hidden. The burrowing is mostly horizontal, just below the sediment surface. Deep vertical burrows, common in later eras, are rare. Life hasn't yet learned to fully exploit the third dimension of the sea floor. What strikes me most about this Ediacaran world is its gentleness. There are no predators here, at least none that we would recognize. No creature has jaws or claws or stingers. Nothing chases, nothing flees. The fastest movement is the slow pulsing of medusoid forms through the water. It is a world without fear, where the biggest challenge is finding enough food and avoiding being buried by sediment. The very structure of the ecosystem is different from what will come later. Energy flows from the sun to photosynthetic microbes to the grazers and filter feeders. But there are no secondary consumers, no creatures that eat other large creatures. The food web is simple, almost linear. Everyone is a vegetarian in this peaceable kingdom. I spend days drifting through different Ediacaran environments, each with its own community of strange forms. In deeper water, away from the photosynthetic mats, I find forests of rangermorphs, creatures built on a fractal body plan that maximizes surface area. Some stand upright like Charnia. Others lie flat on the seafloor, their fronds spread wide to capture whatever nutrients the water carries. In very shallow water, almost in the intertidal zone, I discover Kimberella, a creature that hints at changes to come. It's only about 4 inches long, shaped like a flattened dome, with what might be a muscular foot on its underside. Unlike the other Ediacaran fauna, Kimberella seems to have a clear front and back, and it leaves distinctive scratch marks on the microbial mats where it feeds. Some scientists in your time think it might be an early mollusk, a distant ancestor of snails and slugs. If so, it represents a new kind of body organization. Bilateral symmetry with a definite head end. But even Kimberella, advanced as it may be, lacks the features that will soon revolutionize life. It has no shell, no hard parts of any kind. Its body is soft, vulnerable, unprotected. It has no eyes, navigating by chemical senses alone. It moves slowly, feeds slowly, lives slowly in this slow world. As I observe this peaceful Ediacaran ecosystem, I'm aware that I'm witnessing the calm before the storm. These creatures have had the seas to themselves for perhaps 30 million years, evolving their strange forms in the absence of predation, in a world where the biggest advantage is simply being large enough to avoid being buried or swept away. But change is coming. In the chemistry of the seawater, in the oxygen levels, in the types of microbes proliferating in the margins, I can sense the approaching revolution. Life is about to discover possibilities that will make this gentle world seem like a half-remembered dream. The first signs are subtle. Here and there, I notice small creatures with bodies divided into repeated segments. They're tiny, less than an inch long, but their segmentation represents a new kind of organization. Each segment can specialize, can be modified for different functions. It's a modular approach to bodybuilding that will prove incredibly versatile. I also observed the first experiments with mineralization. Some microbes are secreting tiny scales of calcium carbonate, perhaps as waste products, perhaps for protection from ultraviolet radiation. The scales are microscopic, hardly worthy of notice, but they represent a fundamental innovation. The ability to build with minerals, to create hard structures that persist after death. In the sediment, I find traces of new behaviors, burrows that dive deeper, that show more complex patterns. Something is learning to exploit the resources buried in the seafloor, to hide from what? There are no predators yet, but perhaps these early burrowers sense what is coming, preparing defenses for dangers that don't yet exist. The Ediacaran fauna, successful for so long, are beginning to decline. In some areas, their fossil impressions become rarer, replaced by the trails and burrows of more active creatures. It's not a sudden extinction, more like a gentle fading, like a dream upon waking. The quilted, fractal forms that dominated for millions of years are giving way to something new. I position myself at the boundary between the Ediacaran and Cambrian periods, 541 million years ago. It's an arbitrary line, defined by scientists finding different fossils above and below. But as I cross this threshold, I can feel the change. The water seems more energetic, filled with microscopic larvae and eggs. The sea floor shows more disturbance, more evidence of active creatures. The peaceful garden is about to become a jungle. And in this moment of transition, I witness something that will change the world forever. The first shell is so small, I almost miss it. A tiny creature, no larger than a grain of rice, secretes a delicate covering of calcium carbonate. The shell is simple, just a conical cap, like a minute ice cream cone. But it represents a revolution. For the first time in Earth's history, life has learned to build portable armor. I observe this pioneer, a creature your scientists will call Cloudina, as it goes about its daily existence. It's a filter feeder, extending soft tentacles from the opening of its shell to capture microscopic prey. When threatened, though what threatens it in this predator-free world, it can withdraw completely into its protective covering. The shell itself is a marvel of biological engineering. Layer by layer, specialized cells secrete calcium carbonate in an organic matrix. The result is stronger than pure mineral, lighter than solid rock. It's like nature has discovered architecture, creating structures that provide maximum protection with minimum weight. But why now? Why after 3 billion years of soft-bodied life, do shells suddenly appear? The answer lies partly in ocean chemistry. I can sense the difference in the water around me. Calcium levels have risen, the result of increased weathering on land and changes in ocean circulation. Carbon dioxide levels in the atmosphere have dropped, making it easier for organisms to precipitate carbonate minerals. The raw materials for shell building are more available than ever before. Yet chemistry alone doesn't explain the timing. Conditions have been suitable for shell building for millions of years. No, the real driver is about to become clear as I observe what happens next. A soft-bodied creature, something like a primitive worm, approaches the shelled Cloudina. In the Ediacaran, this would have been a peaceful encounter, two creatures ignoring each other, or at most competing for space. But this worm does something unprecedented. It attacks. The attack is clumsy by later standards. The worm has no specialized feeding apparatus, just a simple mouth. It tries to engulf the Cloudina, to digest it whole. Against a soft-bodied creature, this would work, but the shell changes everything. The worm gnaws futilely at the hard covering, unable to reach the soft tissue within. After several minutes, it gives up and moves on. I've just witnessed one of the first acts of predation in the history of complex life, and I've seen the first successful defense. The evolutionary arms race has begun. Word of this innovation seems to spread through the ecosystem like wildfire, though of course it's simply natural selection operating at unprecedented speed, within what seems like geological moments, perhaps a few 100,000 years. Shells are everywhere. I watch as different lineages independently discover the secret of biomineralization. Some build with calcium carbonate like Cloudina. Others use calcium phosphate, creating shells with different properties. Still others mix organic materials with minerals, creating composite structures that combine flexibility with strength. The variety of shell forms that appear is staggering. Conical shells, spiral shells, bivalved shells that hinge open and closed. Some creatures build tubular shells they can extend as they grow. Others create segmented shells that allow flexibility. Each design represents a different solution to the same problems: protection, support, and mineral storage. I follow one particular lineage as it experiments with shell design. These are early mollusks, descendants perhaps of creatures like Kimberella. Their first shells are simple caps, like flattened cones. But over generations, I watch the shells become more elaborate. Some develop internal partitions for strength. Others evolve spiral shapes that provide more internal volume with less material. The spiral shells prove particularly successful. They're strong, efficient, and can grow indefinitely by adding new material at the opening. But shells aren't just for defense. I observe creatures using their hard parts in ways that would have been impossible for soft-bodied organisms. Some use their shells as anchors, cementing themselves to rocks to resist currents. Others use them as digging tools, burrowing into sediment with an efficiency their soft-bodied ancestors could never achieve. Most remarkably, some creatures are learning to use minerals for offense. I watch an early arthropod, perhaps an ancestor of Anomalocaris, that has developed hardened grasping appendages. They're not true shells, but mineralized extensions of its exoskeleton. It uses them to crack open the shells of other creatures, turning defense into vulnerability. This triggers an escalation in shell design. Simple conical shells give way to more elaborate defenses. Some creatures evolve shells with long spines that make them difficult to grasp. Others develop thick, rounded shells that resist crushing. Still others create shells with narrow openings that can be sealed with a mineralized door called an operculum. I'm witnessing evolution in fast forward. What once took millions of years now happens in thousands. The presence of predators has changed the pace of natural selection. Every improvement in offense drives improvements in defense, which drives further improvements in offense. It's an arms race that will continue for the next 500 million years. The appearance of shells has another profound effect. It changes the seafloor itself. Soft-bodied creatures leave few traces when they die, their tissues quickly consumed by bacteria. But shells persist. I watch as the first shell beds begin to form, accumulations of hard parts that create new habitats. These shell accumulations are colonized by other organisms. Soft-bodied creatures find shelter in the spaces between shells. Filter feeders attach themselves to stable shell surfaces. What begins as death becomes the foundation for new life. The seafloor is becoming three-dimensional in a way it never was before. The Ediacaran fauna, those strange quilted creatures that dominated for so long, cannot compete in this new world. They have no shells, no protection against the new predators. More fundamentally, they seem unable to evolve shells. Whatever their body plan, and scientists in your time still debate what they actually were, it doesn't allow for biomineralization. I watch sadly as the last Dickinsonia in this area is attacked by a pack of small arthropods. They're each less than an inch long, but they have sharp mouthparts and work together. They tear through Dickinsonia's unprotected tissue, reducing the 4-foot giant to scattered fragments in hours. It's a scene that would have been impossible just 1 million years earlier. Yet even as the Ediacaran fauna fade, I'm amazed by the diversity that replaces them. Every possible variation on shelled life appears. Some creatures build external shells like snails. Others develop internal shells for support, like the pen of a squid. The arthropods take a different approach, developing a mineralized exoskeleton that covers their entire body. I spend time observing the Trilobites, perhaps the most successful of these early arthropods. Their name means three-lobed, referring to the 3 longitudinal sections of their body, but it's their hard exoskeleton that makes them revolutionary. Made of calcite reinforced with organic compounds, it provides protection while still allowing movement at the joints. A typical trilobite of this early period is about 2 inches long, though some species are already growing larger. The exoskeleton is divided into segments that allow the animal to roll into a ball for protection, like a pillbug. The head shield often bears spines or other ornamentations, each species with its own distinctive pattern. But trilobites have evolved something even more remarkable than armor. As I examine one closely, I notice structures on its head shield that catch the light differently. These are eyes. The first true compound eyes in the fossil record, representing perhaps the most revolutionary innovation in the history of life. Let me draw you deeper into this miracle of biological engineering. Each compound eye is a universe of precision, containing hundreds of individual optical units called ommatidia. I position myself close enough to examine a single ommatidium, marveling at the complexity packed into a structure smaller than the head of a pin. At the surface lies a lens made of pure calcite, a mineral that trilobites have learned to grow with crystallographic perfection. This is not the chaotic calcite of a limestone cave, but ordered crystal with its optical axis precisely aligned to minimize double refraction. The trilobite cells have become master crystallographers, secreting calcium and carbonate ions in exact ratios, controlling temperature and chemistry to produce lenses clearer than the finest optical glass your civilization will create. But creating a perfect lens is only the beginning. Beneath each lens lies a sophisticated optical system. The lens focuses incoming light onto a transparent crystalline cone that acts as a light guide, channeling photons down through the ommatidium. This cone is made of layers of protein with slightly different refractive indices, creating what optical engineers in your time would recognize as a graded index waveguide, preventing light from scattering as it travels deeper into the eye. At the base of each ommatidium sits the photoreceptor, a cluster of cells arranged like the segments of an orange. Each cell extends a thin projection, a rhabdomere, containing thousands upon thousands of rhodopsin molecules. These are the same light-sensitive proteins that will enable vision in every animal that follows, from you to the smallest insect. But here, in these ancient seas, rhodopsin is being utilized for the first time to transform photons into the electrical signals of neural communication. The chemistry of sight is exquisitely complex. When a photon strikes a rhodopsin molecule, it causes a conformational change that triggers a cascade of biochemical reactions. A single photon can generate the movement of millions of ions, amplifying the signal until it becomes strong enough to fire a neuron. This quantum mechanical sensitivity, the ability to detect individual particles of light, has been achieved by biological systems hundreds of millions of years before your physicists will prove that such sensitivity is theoretically possible. The electrical signals from each ommatidium travel along neural pathways toward processing centers in the trilobite's brain. Here, the real magic of vision begins. The brain must take hundreds of individual signals, each ommatidium providing one pixel of information, and combine them into a coherent image of the world. I observe this neural processing in action as a trilobite encounters a small worm moving across the seafloor. The movement registers as a sequential activation of ommatidia, creating a wave of neural activity across the eye. The trilobite's brain compares the timing of these signals, calculating not just that something is moving, but its direction, speed, and approximate size. Neurons that fire together become linked in processing networks, building up complex representations of visual scenes. But this trilobite's eyes are sophisticated beyond simple motion detection. Different ommatidia are optimized for different functions. Those at the center of the eye have narrower acceptance angles, providing higher resolution for examining objects directly ahead. Those at the periphery have wider angles, better for detecting movement in the trilobite's peripheral vision. The eye is specialized from center to edge, each region tuned for its particular role. The trilobite can also perceive polarized light, though this ability is largely invisible to most modern animals. Sunlight becomes polarized as it passes through water, creating patterns that change predictably with the sun's position. By detecting these polarization patterns, the trilobite can navigate even when the sun itself is obscured by clouds, or when it's swimming in murky water. It's a biological compass of extraordinary sophistication. Color vision, too, has appeared in these ancient seas. This trilobite's rhodopsin proteins exist in several varieties, each sensitive to different wavelengths of light. Some respond best to the blue-green light that penetrates deepest into water. Others are tuned to shorter wavelengths, detecting the violet light that filters through the clear tropical seas. Still others respond to longer wavelengths, the green and yellow light that reveals the photosynthetic organisms that form the base of the food web. The evolution of color vision transforms the trilobite's world from shades of brightness and darkness to a rich palette of hues. A potential meal can be distinguished from a toxic organism by color alone. A mate can be identified by subtle color patterns invisible to predators with simpler visual systems. The seafloor becomes a canvas of information, each color and pattern carrying meaning for those equipped to read it. But perhaps most remarkably, the trilobite has evolved the neural circuitry to process all this visual information in real time. As it moves across the seafloor, its brain continuously updates its three-dimensional map of the environment. Objects that disappear behind obstacles are remembered and tracked. Movements are predicted and anticipated. The visual world becomes not just a stream of sensory data, but a coherent, persistent reality. I watch as this trilobite demonstrates the power of its visual system. A predator approaches from the right, still 50 feet away. The trilobite's compound eyes detect the movement instantly, analyzing the predator's size, speed, and trajectory. Neural circuits inherited from blind ancestors are now guided by precise visual information. The trilobite doesn't just flee randomly, it calculates the best escape route, heading for a rocky outcrop that will provide both physical protection and visual concealment. The sophistication of trilobite vision varies enormously among species. Some deep water forms have eyes enlarged to gather every precious photon in the dim depths. Their lenses are larger, their neural processing more sensitive, their behavior adapted to a world where light is scarce but precious. Other species, living in bright shallow waters, have evolved eyes optimized for acute resolution, capable of detecting the finest details of prey, predators, and potential mates. Some trilobites have taken eye evolution to extraordinary extremes. Certain species have developed eyes on stalks that can swivel independently, providing a nearly complete panoramic view of their surroundings. Others have evolved eyes with different focal distances, some for near vision, and others for spotting distant movements. A few species have even developed pinhole eyes alongside their compound eyes, trading light-gathering ability for the incredibly sharp focus that only a pinhole aperture can provide. The development of trilobite eyes from embryo to adult reveals the genetic programs that control this complex construction. I observe a juvenile trilobite in the process of molting, its new exoskeleton still soft and flexible. As the exoskeleton hardens, new ommatidia are added to the eyes, increasing their resolution and sensitivity. The number and arrangement of ommatidia is controlled by ancient genes that will later be discovered to control eye development across the entire animal kingdom. These same genetic programs, refined over millions of years of evolution, contain the instructions for building the precisely oriented calcite lenses, the light-guiding crystalline cones, the photosensitive rhabdomeres, and the neural networks that process visual information. They represent a library of information accumulated through countless generations of natural selection. Each gene a lesson learned about how to see. The evolution of trilobite vision has profound ecological consequences. With acute eyesight comes the ability to be an active predator rather than a passive filter feeder. Trilobites can now hunt specific prey, choosing the most nutritious or easiest targets while avoiding dangerous or toxic organisms. This selective predation pressure drives the evolution of new defensive strategies in prey species, accelerating the evolutionary arms race that characterizes the Cambrian explosion. Vision also revolutionizes social behavior. Trilobites can now recognize members of their own species by sight, leading to the evolution of visual signals for communication. Some species develop distinctive color patterns that identify them to potential mates while warning competitors. Others evolve elaborate courtship displays, using movement and posturing to demonstrate their fitness to choosy partners. The mating behaviors I observe are surprisingly sophisticated. A male trilobite approaches a female with a characteristic dance, moving in spirals around her while displaying the bright patches of color on his exoskeleton. His compound eyes are locked on hers, monitoring her response to his display. She assesses not just his colors, but the precision of his movements, the symmetry of his body, the condition of his armor. Vision has made mate choice visual, adding new dimensions to sexual selection. But trilobite eyes are not perfect. They have limitations that will constrain their evolution for the remainder of their long history. The compound eye design provides wide-angle vision, but limited resolution compared to the camera eyes that mollusks are simultaneously developing. The calcite lenses, while crystal clear, are difficult to adjust for focusing, limiting trilobites to a relatively fixed visual range. These limitations drive the evolution of behavioral adaptations. Trilobites learn to position themselves at optimal distances from their prey, where their eyes function most effectively. They develop hunting strategies that maximize the advantages of their wide-field vision while minimizing the disadvantages of limited resolution. They become masters of movement detection while remaining relatively poor at discerning fine details. As I observe this trilobite going about its daily existence, I'm struck by how vision has transformed not just its behavior, but its entire relationship with the world, where its blind ancestors moved through a universe of chemical gradients and tactile sensations. This creature inhabits a world of light and shadow, color and movement, depth and distance. The development of sight has literally illuminated the world, making visible relationships and opportunities that were previously hidden. The compound eyes of this trilobite represent one of evolution's great experiments in perception, a solution to the challenge of seeing that will be refined and perfected by insects and crustaceans for hundreds of millions of years to come. In developing vision, trilobites haven't just gained a new sense. They've opened up entirely new ways of being alive, new strategies for survival, new possibilities for interaction with their environment and each other. This is the miracle I witness in these ancient seas, the birth of sight itself, the moment when life first learned to turn photons into perception, to transform the invisible electromagnetic radiation of the sun into the rich visual experience of seeing. It's a transformation as profound as the origin of life itself, opening up new dimensions of existence that will shape the evolution of animals forever after. With this revolutionary gift of sight now established across many species, the Cambrian seas have become an arena where vision shapes every interaction. I follow a hunting trilobite as it demonstrates this new reality, stalking across the sea floor with purpose that would have been impossible for its blind ancestors. Its compound eyes swivel independently, scanning for movement with the precision of sophisticated optical instruments. It spots a small worm burrowing in the sediment, the motion betraying its presence through subtle changes in light and shadow that only eyes could detect. The trilobite approaches carefully, using its legs to feel the vibrations through the sea floor, while its visual system continues to track the target. At the last moment, it lunges, using specialized appendages near its mouth to grab the worm before it can retreat into its burrow. It's a successful hunt, but I notice the worm has its own defenses. As the trilobite crushes its body, the worm secretes a noxious chemical that makes the predator recoil. It's too late for this worm, but others of its species will benefit. The trilobite will remember this unpleasant experience and may avoid similar worms in the future. Chemical defenses are another innovation of the Cambrian explosion. Unable to build shells, some soft-bodied creatures develop toxins, irritants, or foul-tasting compounds. I observe a soft coral that secretes a milky substance when disturbed. A fish-like creature that attempts to graze on it immediately spits it out and swims away rapidly, shaking its head as if to clear a bad taste. The variety of defensive strategies is matched by the variety of offensive ones. Some predators develop drill-like mouth parts to penetrate shells. Others evolve crushing mandibles. Still others become ambush specialists, hiding in sediment with only their eyes exposed, waiting for unwary prey to pass. I discover a creature that represents perhaps the ultimate predator of this early Cambrian sea. Anomalocaris, meaning abnormal shrimp, though it's neither abnormal nor a shrimp. It's huge by the standards of its time. This specimen is nearly 3 feet long. Its body is streamlined, propelled by undulating fins along its sides. but it's the front end that's truly fearsome. Two great grasping appendages, each over 6 inches long, extend from its head. They're jointed like shrimp arms but more flexible, able to curl inward to bring prey to the mouth. The mouth itself is a circular opening surrounded by plates that can grind and crush. But most remarkably, Anomalocaris has eyes, enormous compound eyes on stalks, each containing thousands of individual lenses. Now let me show you the hunt in all its terrible magnificence. For this is predation refined to an art form that will define marine ecosystems for millions of years to come. Anomalocaris begins its hunt long before any prey comes into view. 30 feet above the sea floor, it maintains a slow, energy-efficient cruise, its undulating lateral fins creating almost no disturbance in the water. These fins are marvels of hydrodynamic engineering, thin membranes supported by flexible rays that can adjust their angle and curvature to provide thrust, steering, or stabilization. The fin motion is hypnotic. Waves of muscular contraction rippling from head to tail, propelling this 3-foot predator forward with silent grace. Its enormous compound eyes, each containing over 10,000 individual lenses, scan the seafloor below in a systematic pattern. Unlike human vision, which focuses attention on specific areas, Anomalocaris processes the entire visual field simultaneously. Movement detectors in its visual cortex respond to even the slightest stirring in the sediment. Object recognition circuits compare shapes and patterns against a library of prey images encoded in its neural networks. Distance calculators use parallax and relative motion to build a 3-dimensional map of the hunting ground. The metabolic cost of operating these sophisticated sensory systems is enormous. Each ommatidium requires a constant supply of energy to regenerate the light-sensitive chemicals that enable vision. The neural processing alone consumes nearly 1/3 of Anomalocaris' resting metabolism. But the investment pays dividends. In a world where blindness means relying on chance encounters with prey, vision provides the decisive advantage of choice. A flicker of movement catches the attention of thousands of ommatidia simultaneously. On the sandy bottom, 60 feet away, a group of trilobites has emerged from the algal mats where they have been feeding. There are 7 individuals, each about 4 inches long, their compound eyes glinting in the filtered sunlight. They move with the cautious alertness of prey animals, but their visual systems, sophisticated as they are, cannot match the processing power dedicated to predation in Anomalocaris' brain. The predator's hunting program activates. Neural circuits that evolve specifically for this moment begin their calculations. Wind speed, water current, distance to prey, escape vectors, energy expenditure. All are weighed in an instant. The trilobites are within optimal striking range. Their position on open sand offers no immediate shelter. The angle of approach will bring Anomalocaris out of the sun, making it harder for the prey to spot the attack until it's too late. Decision made, Anomalocaris begins its approach. But this is not the desperate lunge of a hunger-driven predator. This is calculated precision. The great predator adjusts its buoyancy, using internal gas-filled chambers to become slightly denser, beginning a gradual descent while maintaining forward momentum. Its fin beats slow further, reducing water disturbance. Even its eye movements become more deliberate, tracking the prey while minimizing the light reflections that might betray its presence. At 40 feet distance, one trilobite freezes. Something has triggered its predator detection instincts. Perhaps a shadow moving across its visual field, perhaps a change in the water pressure, as Anomalocaris displaces fluid in its descent. The Trilobite's alarm posture immediately alerts its companions. All 7 creatures tense, their legs poised for rapid escape, their stalked compound eyes swiveling to scan for danger. But Anomalocaris has hunted Trilobites before. It recognizes the alarm behavior and adapts instantly. Its approach angle shifts slightly, taking advantage of a blind spot created by the Trilobite's own visual system. Its great grasping arms, normally held close to its body for streamlining, extend forward and spread wide, creating a capture net nearly 18 inches across. These arms are themselves masterpieces of evolutionary engineering. Each is composed of dozens of articulated segments, making them more flexible than any vertebrate limb. The segments are hollow, containing hydraulic fluid that can be pumped to extend or contract the arm with lightning speed. Along the inner edge of each arm, hundreds of hooked spines point inward, perfect for grasping struggling prey without piercing vital organs. Anomalocaris has learned that damaged prey often releases chemicals that attract competitors or scavengers. The muscles that operate these arms are among the most powerful in the animal kingdom. Cable-like bundles of contractile proteins that can generate tremendous force. But power without control is useless in the delicate art of prey capture. Sensory nerves run throughout each arm, providing continuous feedback about pressure, position, and the texture of whatever is being grasped. The arms can be strong enough to crush shell or gentle enough to hold a soft-bodied prey without injury, depending on the predator's needs. 25 feet from the Trilobite group, Anomalocaris makes its final adjustment. Its fins can forward, acting as brakes to control the descent speed. Its mouth, normally closed to reduce drag, opens to reveal the complex feeding apparatus within. The circular opening is lined with flexible plates that can expand or contract, acting as both grippers and crushers. Behind these plates, rows of smaller teeth help process food before it enters the digestive system. 15 feet, 10 feet. The Trilobites still haven't detected the approaching predator, their attention focused on the wrong area of their visual field. But now it's too late. Anomalocaris strikes with explosive speed, its entire body extending like a coiled spring suddenly released. The muscular contractions that drive the attack begin at the tail and propagate forward in a coordinated wave, each segment adding its power to the thrust. The lateral fins become rigid hydrofoils, channeling water flow to provide maximum acceleration. The attack covers the final 10 feet in less than a second. The Trilobites scatter in all directions, their own reflexes lightning fast by the standards of their time. But 6 of the 7 are outside the strike zone, their escape routes clear. The 7th, a mature female heavy with developing eggs, is directly in the path of Anomalocaris' attack. The grasping arms close around her with practiced precision. The left arm curves inward to block her primary escape route, while the right arm sweeps across to make the actual capture. The hooked spines engage her exoskeleton at dozens of points, distributing the grasping force to avoid cracking the valuable armor. Within milliseconds, the Trilobite is immobilized, held firmly but not crushed in the predator's grip. But the Trilobite's defenses are far from exhausted. As she's lifted from the sea floor, she curls her body into the defensive posture that has served her kind well. The ability to roll into a ball, protecting her soft underside within a sphere of hard exoskeleton. Her head shield locks against her tail section, creating an armored capsule with no obvious points of entry. It's a defense that can deter most predators, but Anomalocaris has encountered this strategy many times before. The predator's response reveals the sophistication of its feeding behavior. Rather than attempting to crush the rolled Trilobite immediately, Anomalocaris brings it close to its head, where specialized sensory organs examine the prey in detail. Chemical receptors taste the water around the trilobite, identifying species, health status, and even the trilobite's recent diet. Pressure sensors in the grasping arms map the exact structure of the curled exoskeleton, identifying potential weak points. Most remarkably, Anomalocaris begins to manipulate its prey systematically, rotating the ball to examine it from every angle. The predator has learned that trilobite exoskeletons, while strong, are not uniformly armored. The joints between segments, designed for flexibility during molting, remain slightly weaker. The ventral surface, normally protected when the animal feeds on the bottom, may have thinner plating in some species. Each individual trilobite's armor reflects its age, genetics, diet, and molting history. And Anomalocaris has evolved the sensory capability to read these subtle differences. After nearly a minute of this examination, Anomalocaris makes its choice. The trilobite's left side shows subtle signs of damage from a previous encounter, perhaps with another predator, or from a difficult molting. The exoskeleton there is slightly irregular, creating a potential entry point. The predator positions its prey carefully, aligning the weak spot with its circular mouth. What follows is not the crude crushing that might be expected, but a demonstration of precision feeding that rivals the techniques of modern surgical predators. Anomalocaris' mouthplates work like a sophisticated toolkit, some providing steady pressure while others probe and manipulate. The predator applies force gradually, feeling for the exact moment when the exoskeleton begins to yield. The first crack is almost inaudible, but Anomalocaris' sensitive feeding apparatus detects it instantly. More pressure is applied, not randomly, but targeted precisely at the point of failure. The crack propagates slowly along the natural stress lines in the trilobite's exoskeleton. A second crack appears, then a third. The predator is essentially performing controlled demolition, using the trilobite's own structural weaknesses against it. As the armor fails, Anomalocaris' feeding behavior shifts again. The mouthplates that were applying crushing pressure now become delicate manipulators, carefully separating the broken exoskeleton to expose the soft tissues within. The trilobite, still alive but unable to escape, makes a final attempt at chemical defense, releasing bitter compounds from specialized glands. But Anomalocaris' digestive system has evolved countermeasures to these toxins, producing neutralizing enzymes that render most chemical defenses ineffective. The feeding process that follows is methodical and efficient. Anomalocaris doesn't simply tear into its prey, but carefully removes organs and tissues in a specific order. The digestive glands come first, rich in enzymes and nutrients. The reproductive tissues follow, packed with proteins essential for the predator's own reproduction. The muscle blocks are consumed next, providing the bulk of the meal's energy content. Even the nervous system is not wasted, its lipid-rich tissues providing essential fatty acids. Throughout this process, Anomalocaris maintains its position in the water column, its lateral fins making subtle adjustments to counteract the predator's movements as it feeds. The entire feeding sequence takes nearly 10 minutes, far longer than the capture itself, but essential for extracting maximum nutrition from each kill. In the energy-expensive world of active predation, waste is a luxury that Anomalocaris cannot afford. As the feeding concludes, Anomalocaris faces the final challenge of successful predation, dealing with the indigestible remains. The trilobite's exoskeleton, now emptied of nutritious content, cannot be fully consumed. But even this waste is managed efficiently. The predator's mouthplates compact the shell fragments into a neat package, which is then expelled. These compressed shell packages will become part of the fossil record, providing future scientists with direct evidence of Anomalocaris' feeding behavior. With the hunt completed, Anomalocaris enters a post-feeding phase that is as carefully orchestrated as the hunt itself. Its metabolism shifts to focus on digestion, with blood flow redirected to the gut and digestive enzymes flooding the stomach. The predator's activity level drops as energy is diverted from swimming muscles to the complex biochemical processes that will extract every possible nutrient from the meal. But even in this satisfied state, Anomalocaris remains alert. Its visual system continues to scan the environment, now looking not for prey, but for potential competitors who might be attracted by the chemical traces of the recent kill. Other Anomalocaris individuals, smaller predators seeking scraps, scavengers hoping for leftovers, all pose potential threats to a temporarily sluggish, well-fed predator. This single hunt, from initial detection to final disposal of remains, represents the culmination of millions of years of evolutionary refinement. Every aspect of Anomalocaris' anatomy and behavior has been shaped by the demands of active predation. Its hydrodynamic body form, its sophisticated sensory systems, its specialized feeding apparatus, its complex behavioral programs, all serve the singular purpose of finding, capturing, and consuming other animals. But perhaps most significantly, this hunt demonstrates how predation has become a driving force in the evolution of all marine life. The Trilobites' defensive behaviors, its chemical deterrence, its armored exoskeleton, all evolved in response to predation pressure. The other Trilobites that escaped will carry the memory of this attack, becoming warier, faster, better at detecting approaching predators. Their offspring will inherit not just their genes, but their learned behaviors, continuing the endless arms race between predator and prey. As I watch Anomalocaris swim away from the hunt site, its belly full and its energy reserves replenished, I'm witnessing more than just successful predation. I'm seeing the engine that drives the Cambrian explosion itself, the constant pressure of life feeding on life, creating an ever-escalating spiral of innovation, complexity, and diversity. In learning to hunt, Anomalocaris has not just secured its own survival, but accelerated the evolution of every creature that shares these ancient seas. Yet even this supreme predator is not invulnerable. As it feeds, I notice scars on its body, healed wounds from past encounters, perhaps from others of its kind, competing for territory or mates, or perhaps from prey that fought back more successfully than this trilobite. The arms race ensures that no advantage is permanent, no predator is perfect. The impact of predation extends beyond individual encounters. It's reshaping entire ecosystems. Creatures that once lived exposed on the seafloor now burrow for protection. The simple horizontal burrows of the Ediacaran give way to complex three-dimensional tunnel systems. Some creatures dig deep vertical shafts with multiple entrances and emergency exits. I follow one such burrow system, created by a worm-like creature about 8 inches long. The main shaft descends 18 inches into the sediment, with horizontal galleries branching off at different levels. Each gallery has multiple connections to the surface, allowing rapid escape if a predator enters one opening. The walls are reinforced with mucus that hardens into a tough lining. This increased burrowing has profound effects on ocean chemistry. As creatures mix the sediment, they bring oxygen to deeper layers, allowing bacteria to break down organic matter more efficiently. nutrients are recycled faster. The seafloor becomes more productive, supporting more life. What began as hiding from predators ends up enriching the entire ecosystem. The Cambrian explosion is often portrayed as a time of fierce competition, nature red in tooth and claw. But I observe as much cooperation as competition. Creatures form symbiotic relationships for mutual benefit. Small fish-like creatures follow larger predators, feeding on scraps. Certain worms live in the discarded shells of mollusks, keeping them clean in exchange for shelter. I discover a remarkable partnership between a brachiopod and a species of algae. The brachiopod, a shelled creature superficially similar to a clam, has evolved transparent windows in its shell. Algae live beneath these windows, protected from grazers while still able to photosynthesize. In return, they provide oxygen and nutrients to their host. It's an elegant solution that benefits both parties. As I survey this transformed world, I'm struck by how different it is from the peaceful Ediacaran seas. Where once there was simplicity, now there is complexity. Where once there was peace, now there is dynamism. Life has discovered competition and cooperation, offense and defense, vision and camouflage. The quiet garden has become a bustling marketplace of evolutionary innovation. And yet, for all the changes, this is still just the beginning. The Cambrian explosion has more surprises in store. Allow me to tell you the story of a single lineage, for in following one family line through the depths of time, we can understand how the Cambrian explosion played out in the lives of real creatures. I will trace the descendants of a small trilobite I have named Olenoides primalis, following her genetic legacy through 10 million years of evolution. I begin 520 million years ago, in the warm, shallow seas of what will one day be Western North America. Here, in water no deeper than 100 feet, I first encounter Olenoides primalis. She is not impressive by the standards of her time, barely 1.5 inches long, with a relatively simple exoskeleton divided into 13 segments. Her compound eyes are small, containing only about 50 ommatidia each, enough to detect movement and judge distance, but lacking the sophisticated processing power of larger predators. But what Olenoides lacks in size and visual acuity, she makes up for in adaptability. Her species has evolved a generalist lifestyle, equally comfortable burrowing in soft sediments or scavenging on hard surfaces. Her mouthparts can process both algal matter and small animal prey. Her respiratory system functions efficiently in both well-oxygenated surface waters and the lower oxygen conditions of sediment burrows. She is evolution's equivalent of a Swiss Army knife, not the best at any one thing, but capable of doing many things well enough to survive. I watch as Olenoides primalis mates with a male slightly smaller than herself. Trilobite reproduction at this time is still relatively simple. The male approaches carrying packets of sperm, which he deposits near the female's genital openings. The female then releases hundreds of tiny eggs into the water, where external fertilization occurs. Most of these eggs will be eaten by planktonic predators, but enough survive to perpetuate the species. The larvae that hatch from these eggs face a world very different from that of their parents. They are protaspids, tiny shield-like creatures, less than 1 millimeter across, with no body segments and only the most basic of sensory organs. They drift in the plankton for several weeks, feeding on bacteria and microscopic algae. It's a perilous existence. Storm waves can wash them into unsuitable environments. Predators like early jellies can swallow them by the thousands. Changes in water chemistry can kill entire populations, But those that survive this planktonic phase undergo a remarkable transformation. Through a series of molts, they gradually develop the segmented body plan of adult trilobites. With each molt, new segments are added, new sensory organs develop, new capabilities emerge. The process takes nearly a year, but those juveniles that complete it successfully have earned their place in the adult community. One such survivor, a descendant of Olenoides primalis, emerges from its final juvenile molt with a genetic mutation that will prove significant. A small change in the gene that controls eye development has resulted in compound eyes with 15% more ommatidia than the parental generation. It's a subtle difference, barely noticeable to casual observation, but it provides a measurable advantage in detecting both prey and predators. This enhanced vision proves valuable in the increasingly complex ecosystems of the Middle Cambrian. As predators become more sophisticated and prey more elusive, the ability to see slightly farther, to detect movement slightly sooner, to resolve details slightly better, can mean the difference between successful feeding and starvation, between escaping predation and becoming someone else's meal. Over the next million years, I watch as this enhanced vision mutation spreads through the Olenoids population. Individuals with better eyesight are more successful at finding mates, avoiding predators, and raising offspring. The average number of ommatidia per eye increases from 50 to 75. The visual processing regions of the trilobite brain expand to handle the increased data flow. The species is evolving before my eyes. But evolution is not a straight line, and the descendants of Olenoides primalis face new challenges as their environment changes. Sea levels fluctuate, altering the depth and chemistry of their habitat. New predators evolve, requiring new defensive strategies. Climate shifts change the distribution of food sources. Each challenge eliminates some individuals while favoring others, continuously sculpting the species. 515 million years ago, I witnessed a major crisis in the Olenoids lineage. A period of oceanic turnover brings cold, oxygen-poor water into their shallow habitat. Many trilobite species go extinct during this event, unable to cope with the changed conditions. But the Olenoids lineage survives, though barely. The population crashes to perhaps a few hundred individuals scattered across a handful of refugia. This near extinction becomes a turning point in the lineage's evolution. With the population so severely reduced, genetic drift becomes a powerful force. Mutations that might have been diluted in a large population become fixed in these small survivor groups. When conditions improve and the population expands again, it does so from a genetically narrow base that differs significantly from the ancestral species. The post-crisis Olenoids show several new features. Their exoskeletons have become noticeably thicker, providing better protection against the crushing attacks of evolving predators. Their burrowing ability has improved, with stronger digging appendages and more efficient gill systems for extracting oxygen from sediment-poor waters. Most remarkably, they've developed the ability to remain buried in sediment for extended periods, entering a kind of suspended animation during unfavorable conditions. These adaptations prove crucial as the Middle Cambrian progresses. The seas become increasingly dominated by large predators. Anomalocaris and its relatives patrol the water column, while smaller but equally dangerous arthropods hunt in the sediments. For a small, relatively defenseless creature like Olenoides, the ability to hide becomes more valuable than the ability to fight. I observe the development of increasingly sophisticated burrowing behaviors. Early generations of post-crisis Olenoids dug simple vertical tubes, barely adequate for temporary concealment. But as predation pressure intensifies, their burrow architecture becomes more complex. U-shaped burrows with multiple entrances allow rapid escape if discovered. Deeper burrows provide access to the rich organic matter buried in older sediments. Branching burrow systems create three-dimensional mazes that confuse pursuing predators. The evolution of complex burrowing drives changes in Olenoides' morphology that extend far beyond simple digging adaptations. Their bodies become more streamlined for movement through sediment. Their sensory systems adapt to the chemical-rich environment of the seafloor. Their digestive systems become more efficient at processing the organic detritus that forms their primary food source in burrow environments. 510 million years ago, a significant development occurs in the Olenoids lineage. A population living in deeper water, beyond the reach of most visual predators, begins to evolve in a different direction from their shallow water relatives. These deep water forms develop enlarged eyes with more sensitive photoreceptors, able to detect the faintest traces of bioluminescence in the abyssal darkness. Their bodies become more delicate, less armored, as they adapt to a world where concealment is more important than physical defense. Meanwhile, the shallow water Olenoids populations face different selective pressures. Here, the arms race between predator and prey continues to escalate. I watch as successive generations develop increasingly elaborate defensive features. Some develop long spines that make them difficult to swallow. Others evolve chemical defenses, secreting toxic or distasteful compounds that make predators avoid them. Still, others become masters of camouflage, their exoskeletons matching the color and texture of their preferred habitats so precisely that they become nearly invisible. But perhaps the most remarkable adaptation I observe is the evolution of schooling behavior in certain Olenoids populations. These trilobites have learned to coordinate their movements, forming large aggregations that move across the seafloor like underwater herds. When threatened, they can quickly form defensive formations, with the largest individuals on the outside protecting the smaller ones within. It's a strategy that will be reinvented countless times in the history of life. But here in the Cambrian, it's a revolutionary innovation. The genetic basis of this schooling behavior is complex, involving changes in sensory systems, neural processing, and behavioral programming. Trilobites must be able to detect and respond to the chemical signals and movement patterns of their neighbors. They must suppress their individual feeding instincts when the group decides to move. They must be able to recognize members of their own species, while excluding closely related but competing forms. By 505 million years ago, the Olenoids lineage has diverged into at least 6 distinct species, each adapted to different environments and lifestyles. There's the original shallow water form, now heavily armored and equipped with sophisticated chemical defenses. There's the deep water form with its enormous eyes and delicate build. There are the schooling forms that dominate the open sea floor in vast aggregations. There are specialized burrowing forms that rarely emerge into the water column at all. But the most remarkable member of the lineage is Olenoides magnificus, a giant form that has evolved in the nutrient-rich waters near ancient reef systems. These trilobites grow to over 8 inches in length, making them among the largest arthropods of their time. Their success is based on a unique feeding strategy. They've learned to cultivate gardens of algae on their own exoskeletons, creating portable ecosystems that provide both food and camouflage. The algae-trilobite partnership is a marvel of evolutionary innovation. Special grooves and chambers in the exoskeleton provide optimal growing conditions for the algae, which receive protection and nutrients from their mobile host. In return, the trilobites harvest the algae for food and benefit from the oxygen they produce. The algae also help camouflage the trilobites, breaking up their outline and making them harder for predators to detect. This symbiotic relationship requires sophisticated biochemical coordination. The trilobites must produce the right nutrients to feed their algal partners without poisoning them. They must maintain optimal pH and oxygen levels in their cultivation chambers. They must time their harvesting to maximize algal productivity while ensuring the survival of their biological gardens. As I follow the Olenoids lineage toward the end of the Cambrian, I witness both triumph and tragedy. The diverse array of species represents a remarkable adaptive radiation, with forms specialized for every conceivable marine niche. But the very success of the lineage makes it vulnerable to environmental change. When sea levels begin to drop at the end of the Cambrian, many of their specialized habitats disappear. The deep water forms are the first to suffer, as oceanic circulation changes reduce the oxygen content of deep basins. The reef-associated forms decline as changes in water chemistry stress the reef-building organisms they depend upon. The schooling forms find their coordinated behavior less effective as prey becomes scarcer, and predator strategies evolve to exploit group vulnerabilities. But the burrowing forms, those humble descendants of the crisis survivors, prove remarkably resilient. Their ability to retreat into sediment, to wait out unfavorable conditions, to survive on minimal resources, serves them well during this time of environmental stress. They become the foundation for the trilobite radiations of the following Ordovician period. 510 million years after the journey began, I find myself observing the latest generation of Olenoids descendants. They bear little resemblance to their ancestor, Olenoides primalis. They're larger, more heavily armored, with sophisticated sensory systems and complex behaviors. Yet, at the molecular level, they carry the same basic genetic toolkit, modified and elaborated, but fundamentally unchanged. The story of the Olenoids lineage is the story of the Cambrian explosion itself in microcosm. It shows how a single, relatively simple ancestor can give rise to a remarkable diversity of forms through the gradual accumulation of small changes. It demonstrates how environmental challenges can both eliminate species and create opportunities for innovation. It illustrates how the interaction between organisms and their environment drives the endless creativity of evolution. But perhaps most importantly, it reveals how contingent evolutionary history truly is. The survival of Olenoids primalis's lineage through the mid-Cambrian crisis was largely a matter of chance. Being in the right place at the right time, with the right combination of genes. Had that crisis been slightly more severe, had the refugia been slightly less suitable, had the genetic variation been slightly different, the entire subsequent radiation might never have occurred. This contingency doesn't diminish the wonder of what actually happened. If anything, it makes the story more remarkable. Out of countless possible evolutionary pathways, this particular sequence of events led to this particular array of magnificent creatures. The Olenoids lineage represents just one thread in the vast tapestry of Cambrian evolution, but following that thread reveals the intricate patterns that connect all life. As I prepare to leave the Olenoids lineage at the end of the Cambrian, I'm struck by the realization that their story continues. The burrowing forms that survive the end Cambrian changes will give rise to new radiations in the Ordovician. Some of their descendants will survive the great Ordovician extinction, the Devonian crisis, even the massive Permian catastrophe. The last trilobites won't disappear until the end of the Permian. nearly 300 million years after Olenoides primalis first walked the Cambrian seafloor. In following this single lineage through 10 million years of Cambrian evolution, I've witnessed the power of life to adapt, to diversify, to persist in the face of constant change. The Olenoid story is ultimately a story of resilience, the resilience of genes that can be endlessly recombined, of bodies that can be endlessly reshaped, of behaviors that can be endlessly modified to meet new challenges. This is the deeper meaning of the Cambrian explosion, not just the sudden appearance of complex forms, but the establishment of evolutionary processes that would continue to generate complexity for the remainder of Earth's history. In the lineage of Olenoids, we see evolution learning to be creative, discovering the rules that would govern biological innovation for all time to come. Of all the innovations of the Cambrian explosion, none is more transformative than the evolution of eyes. I position myself in the shallow seas where light penetrates most strongly, to witness this revolution in perception unfold. The first eyes were simple, just patches of light-sensitive cells that could distinguish brightness from darkness. I observed these primitive eye spots in many small creatures. A tiny worm, less than half an inch long, has 2 dark spots on its head. They can't form images, but they can detect the shadow of a predator passing overhead. It's enough to trigger an escape response, diving into the sediment for safety. But evolution doesn't stop at simple light detection. I watch over generations as these eye spots become more sophisticated. Some develop a simple lens to focus light. Others sink into a pit that provides directional information. Each improvement gives its owner a slight advantage, driving the evolution of better and better vision. The compound eyes of arthropods represent one solution to the challenge of seeing underwater. I examine a trilobite eye in detail, marveling at its construction. Each individual lens is a crystal of calcite, oriented precisely to minimize spherical aberration. The lenses are arranged in a hexagonal array, each with its own photoreceptor. Together, they create a mosaic image of the world. Different trilobite species have evolved different eye designs for different lifestyles. Bottom dwellers have eyes on the upper surface of their head shield, looking upward for predators. Active swimmers have eyes on stalks that can swivel independently. Some deep-water species have enormous eyes to gather what little light filters down. Others, living in perpetual darkness, have lost their eyes entirely. I follow a trilobite with particularly well-developed eyes as it navigates its world. Its vision is not like yours. The compound eyes create a pixelated image, with each lens contributing 1 pixel. But it's enough to detect movement, judge distance, recognize shapes. The trilobite can see a predator approaching from 50 feet away, giving it time to escape or defend itself. But compound eyes are not the only solution. I discover early mollusks evolving a completely different type of eye. Instead of multiple lenses, they have a single chamber with 1 lens, more like your own eyes. These camera eyes, as they're called, can potentially form sharper images than compound eyes, though the early versions are still quite simple. I observe a primitive nautiloid, an early cephalopod with a coiled shell. Its eyes are pinhole cameras. No lens, just a small opening that allows light into a chamber lined with photoreceptors. The image is dim and blurry, but it's a true image, not a mosaic. The nautiloid can see the outline of rocks, the shape of prey, the looming form of predators. Vision transforms behavior in profound ways. Creatures that can see can actively hunt rather than waiting for prey to blunder into them. They can choose mates based on visual signals. They can navigate complex environments, avoiding obstacles and finding shelter. The world becomes a visually rich environment rather than a blur of chemical gradients and touch sensations. I witness the beginning of visual communication. A small arthropod, when threatened, spreads its tail fan to reveal eye spots, circular patterns that mimic large eyes. To a predator, it suddenly appears to be facing a much larger creature. It's an illusion, but effective enough to cause hesitation, allowing escape. Colors begin to matter in this visual world. While the deep ocean remains dominated by blues and greens, the only colors that penetrate far through water, shallow areas explode with pigmentation. I observe creatures developing red warning coloration to advertise their toxicity. Others evolve disruptive patterns that break up their outline, making them harder to see against complex backgrounds. Perhaps nowhere is the power of vision more evident than in the success of Anomalocaris. With the largest eyes of any creature in these seas, each compound eye is over an inch across and contains thousands of lenses. It can spot prey from distances that would have been unimaginable in the blind world of the Ediacaran. I swim alongside an Anomalocaris as it patrols its territory. Its eyes are never still, constantly scanning, processing visual information at a rate that must be overwhelming for a nervous system evolved from blind ancestors. It spots a school of small swimmers and immediately changes course to intercept them. The swimmers scatter, but Anomalocaris has already calculated their trajectories. Its grasping arms snatch two of them with precise movements, guided entirely by vision. The evolution of eyes drives the evolution of brains. Processing visual information requires neural complexity far beyond what's needed for chemical senses. I observe the expansion of neural tissue in sighted creatures. The Trilobites develop complex lobes in their head segments dedicated to visual processing. Early vertebrates evolve enlarged front ends of their neural tubes, the beginnings of true brains. But perhaps the most profound effect of vision is how it changes the pace of evolution itself. In a sighted world, appearance matters. Predators can see prey. Prey can see predators. Mates can see each other. Natural selection becomes visual selection. Any mutation that affects appearance is immediately tested against the eyes of others. I observe this visual selection in action among a population of small arthropods. Most are transparent, nearly invisible in the water. But occasionally, a mutation produces one with slight pigmentation. These individuals are more visible to predators and don't survive long. But in a nearby population living among colorful coral-like organisms, pigmentation provides camouflage. There, the transparent individuals are the ones who stand out and get eaten. This visual selection drives rapid diversification. Populations separated by short distances evolve different color patterns, different body shapes, different behavioral displays. The uniform appearance of Ediacaran creatures gives way to endless variety. Every lineage becomes a unique experiment in visual survival. I discover creatures that have evolved beyond simple vision to develop specialized visual systems. Some have eyes sensitive to polarized light, allowing them to see patterns invisible to others. Some can detect ultraviolet wavelengths, opening up a hidden visual channel. Others have eyes specialized for different functions. Large eyes for distance vision, small eyes for close-up work. One particular innovation catches my attention. A group of arthropods has evolved eyes with different types of photoreceptors, sensitive to different wavelengths of light. They can see in color, not the rich color vision you possess, but enough to distinguish between different hues. This color vision provides huge advantages in finding food, avoiding predators, and choosing mates. I watch a color-sighted predator hunting among a coral reef. Where other predators see only shades of blue-gray, this one sees a world of subtle color differences. It can spot the slight yellow tinge of a hidden prey animal, the reddish warning coloration of a toxic species. Its success rate is noticeably higher than that of colorblind competitors. The evolution of color vision drives the evolution of coloration. Creatures that were drab become ornate. Sexual selection, choosing mates based on appearance, leads to elaborate displays. I observe male arthropods developing brilliant patches of color that they flash during courtship. Females evolve the ability to assess these displays, choosing mates with the brightest, most symmetrical patterns. But vision is a double-edged gift. Those who can see can also be seen. The same eyes that spot prey can spot predators. This drives the evolution of camouflage and mimicry to new heights. I discover masters of disguise everywhere I look. Here's a flounder-like creature, though not a true flounder, that has evolved to match the seafloor perfectly. Its skin contains chromatophores, cells that can change color by expanding or contracting pigment-filled sacs. I watch as it swims from a sandy area to a rocky one. Within seconds, its coloration shifts from pale tan to mottled grey and brown. When it settles on the bottom, it becomes nearly invisible. Even more remarkably, some creatures have evolved active camouflage. A small cephalopod demonstrates this ability as I watch. Its skin is covered with thousands of chromatophores under neural control. It doesn't just match its background. It constantly adjusts its pattern as lighting changes, as it moves, as potential threats approach. It's like a living video screen, displaying whatever pattern provides the best concealment. But my favorite visual innovation is bioluminescence. In the deeper waters, where sunlight barely penetrates, creatures have learned to make their own light. I descend into the twilight zone to witness this phenomenon. The darkness is punctuated by flashes and glows. A jellyfish-like creature pulses with waves of blue light that ripple across its bell. The display is hauntingly beautiful, but it serves a purpose. Perhaps to startle predators, perhaps to attract prey, perhaps to communicate with others of its kind. A small crustacean demonstrates a different use of bioluminescence. When threatened, it ejects a cloud of glowing particles, like a luminous smokescreen. The predator attacks the glowing cloud, while the crustacean escapes in the darkness. It's a sacrifice of resources. The luminescent chemicals are metabolically expensive to produce, but better than being eaten. Some predators have evolved their own light-producing abilities. An early deep-sea fish has a luminescent lure dangling from a modified fin ray. The lure glows with an enticing blue-green light, attracting smaller creatures that mistake it for prey. When they approach close enough, the fish's mouth opens with surprising speed, creating a suction that draws them in. The interplay between bioluminescence and vision creates an evolutionary spiral. Better eyes can detect fainter glows, driving the evolution of more sophisticated light displays. More complex displays require better visual processing, driving the evolution of more complex eyes and brains. Each innovation builds on the last. As I return to shallower waters, I reflect on how profoundly vision has transformed life. In just a few million years, the dark world of the Ediacaran has become a realm of light and color, pattern and movement. Creatures that once navigated by touch and chemistry now live in a visually rich environment where appearance can mean the difference between life and death. The evolution of eyes has done more than change how animals perceive the world. it has changed the world itself. The sea floor is now sculpted by creatures seeking visual concealment. The water column is filled with visual signals, flashes of color, threatening displays, camouflage patterns. Even the chemistry of the ocean has been affected, as organisms produce new pigments and luminescent compounds. And this is still early days. The eyes of the Cambrian, remarkable as they are primitive compared to what will evolve later. The compound eyes of trilobites will give rise to the acute vision of mantis shrimps and dragonflies. The simple camera eyes of mollusks will evolve into the sophisticated eyes of octopuses and squids. The light-sensitive patches of early vertebrates will become the color-rich vision of birds and primates. Your own eyes, reading these words, are direct descendants of those first light-sensitive cells that appeared in the Cambrian seas. The gift of sight, once given, can never be taken back. It has shaped every aspect of animal evolution since, from the colors of flowers to the patterns of butterflies to the facial expressions of humans. In the Cambrian explosion, life discovered not just how to see, but how to be seen. It was a revolution that changed everything. 510 million years ago, I find myself in a very special place. The area that will one day be known as the Burgess Shale in the Canadian Rockies. But in the Cambrian, this is not a mountain, but a tropical sea located near the equator. A massive reef complex built by ancient sponges and algae creates a cathedral of limestone rising from the deeper ocean floor. The reef face is nearly vertical, dropping 300 feet into darker waters. At its base, fine mud accumulates slowly, carried by gentle currents from the shallower shelf above. This setting, a steep cliff face adjacent to soft sediments, will prove crucial for preserving one of the most important fossil deposits in Earth's history. I position myself at the top of the reef, in the warm, sunlit waters where life flourishes in extraordinary abundance. The diversity here surpasses anything I've encountered elsewhere in the Cambrian seas. It's as if this reef has become a laboratory for evolutionary experimentation, where life is trying every possible variation on the theme of existence. Swimming past me is Opabinia, a creature so strange that when it was first reconstructed from fossils, scientists thought it must be a joke. It's about 3 inches long with a segmented body and lateral fins for swimming. But its head is what makes it extraordinary. 5 mushroom-shaped eyes protrude from stalks, giving it nearly 360-degree vision. From the front of its head extends a flexible proboscis, like an elephant's trunk, ending in a grasping claw. I watch Opabinia hunt. It swims slowly above the sea floor, its 5 eyes scanning independently for prey. It spots a small worm and descends, its proboscis extending forward. The trunk is remarkably flexible, bending and twisting to follow the worm's evasive movements. Finally, the claw grasps the prey and brings it to Opabinia's mouth, which is located on the underside of its head behind the proboscis. It's a body plan that seems almost whimsical, as if nature were exploring just how strange a successful predator could be. Yet Opabinia thrives here, its unique anatomy perfectly suited to its lifestyle. The 5 eyes provide unmatched visual coverage. The flexible proboscis can reach prey in crevices where rigid grasping arms would fail. It's a reminder that in evolution, strange doesn't mean unsuccessful. Nearby, I encounter Hallucigenia, a creature whose name perfectly captures the bewilderment of scientists trying to understand it. It's about 2 inches long, with a tubular body supported by 7 pairs of long, rigid spines. Along its back are 7 pairs of flexible tentacles, each ending in what might be a grasping structure. For years, scientists reconstructed it upside down, thinking the spines were legs and the tentacles were defensive spikes. Watching Hallucigenia move clarifies its anatomy. It walks on its spines like a living scaffolding, surprisingly stable despite its seemingly precarious structure. The tentacles on its back are indeed feeding appendages, plucking small organisms from the water as it moves. Its head, tiny and easily overlooked, bears simple eyes and a circular mouth lined with teeth. The species here at Burgess seem to be testing the limits of what's possible in animal design. I observe Wiwaxia, covered in scale-like armor and bearing 2 rows of long spines. Is it a mollusk? An annelid worm? Something else entirely? Even watching it alive, its relationships are unclear. It grazes on microbial mats, its body protected by an armor coat that would make a medieval knight envious. But perhaps the most successful group here are the arthropods, represented by dozens of species in every imaginable form. Marella, the most common, is a delicate creature, about an inch long with sweeping head spines and feathery gills. It swims in small schools just above the sea floor, filter feeding on microscopic organisms. I follow a school of Marella as they move in coordinated formation. Their synchronized swimming creates water currents that help bring food particles to the group. When a predator approaches, a larger arthropod with grasping appendages, the school explodes in all directions, confusing the attacker. Most escape, though one unlucky individual is caught. The diversity of arthropods here is staggering. Some, like Liancoilia, have developed specialized grasping appendages that extend far forward of the body. Others, like Burgessia, have evolved broad carapaces that cover most of their body. Each species has found its own solution to the challenges of finding food and avoiding becoming food. Among the most spectacular arthropods is Herdia, a relative of Anomalocaris, but with its own unique features. Nearly a foot long, it has a large head shield with protruding compound eyes. But most remarkably, its front bears a series of comb-like appendages that it uses for filter feeding. It's a gentle giant, cruising through the water with its mouth open, straining out small prey. The reef itself is a complex 3-dimensional habitat. Sponges of various types create the framework, their bodies reinforced with spicules of silica or calcium carbonate. Between the sponges, other organisms find niches. Brachiopods attached to stable surfaces, their shells opening and closing as they filter feed. Primitive echinoderms, ancestors of starfish and sea urchins, cling to the reef with tube feet. I discover gardens of Dynamicius, a creature that defies classification. It looks like a flower, with a cup-shaped body on a long stalk, but is definitely an animal. The cup is lined with feeding appendages that create water currents to capture prey. Is it related to echinoderms, to hemichordates, to something else entirely? Even in life, its affinities are mysterious. In crevices within the reef, I find evidence of ecosystem complexity that rivals modern coral reefs. Small arthropods hide from predators in sponge cavities. Worms bore into the limestone, creating networks of tunnels. Fish-like creatures dart between the sponges, some feeding on smaller animals, others grazing on algae. Speaking of fish-like creatures, I encounter Picaia, a small swimmer about 2 inches long with a distinctive notochord running along its back. It's not impressive compared to the armored arthropods or the bizarre Opabinia, but this humble creature is arguably the most important animal here. It's a primitive chordate, possessing the basic body plan that will eventually give rise to all vertebrates, including humans. I observe Picaia closely. Its body is laterally compressed, with V-shaped muscle blocks that contract in waves to produce swimming motion. The notochord provides a stiff but flexible support for these muscles. It has a distinct head with simple sensory organs and a basic brain. It's a blueprint for all the fish, amphibians, reptiles, birds, and mammals to come. But Picaia is not the only chordate here. I find Metasprigina, another early vertebrate with more advanced features. It has primitive eyes, possible gill arches, and a more developed brain. These early experiments in the vertebrate body plan are happening simultaneously with all the other evolutionary innovations of the Burgess fauna. The predator-prey relationships here have reached new levels of sophistication. I watch a soft-bodied predator that has evolved an extraordinary hunting strategy. It buries itself in the sediment with only its eyes exposed, waiting for prey to pass overhead. When a small arthropod swims by, the predator explodes from the mud, its mouth expanding to create a suction that draws in the prey. It's an ambush strategy that will be reinvented many times in Earth's history. Defense mechanisms are equally sophisticated. Some creatures have evolved chemical defenses so potent that predators learn to recognize and avoid them by sight alone. A soft coral relative produces toxins that cause temporary paralysis. A worm secretes sticky mucus that gums up the mouth parts of attackers. An arthropod can shed its legs when grabbed, leaving the predator with a mouthful of appendages while the body escapes. I'm particularly fascinated by the emergence of cleaning symbioses. Small arthropods have learned to pick parasites and dead tissue from larger creatures. The cleaners get an easy meal, while their clients get relief from parasites. I watch a large trilobite stop swimming and assume a head-down posture. Within moments, several tiny cleaners emerge from hiding and begin working over its exoskeleton. The Burgess Reef represents a pinnacle of Cambrian diversity. By my count, there are representatives of at least 15 major animal groups here, plus numerous creatures of uncertain affinity. Some of these groups will prosper and diversify further. Others will dwindle and disappear, leaving only cryptic fossils to puzzle future scientists. As I explore this ancient reef, I'm aware of the special conditions that will preserve it for posterity. Periodically, earthquakes trigger underwater mudslides that sweep down from the shallow shelf above. These flows carry everything in their path: living animals, empty shells, organic debris, over the reef edge and into the quiet waters below. I witness one such event. The seafloor trembles, and suddenly a wall of mud-laden water cascades over the reef edge. Creatures caught in the flow are instantly buried in fine sediment that excludes oxygen and prevents decay. Even soft tissues are preserved, creating fossils of extraordinary detail. This is how the Burgess Shale will form. Countless burial events over millions of years, preserving snapshots of this incredible ecosystem. Scientists in your time will split open these rocks and marvel at the strange creatures revealed. Many preserved in such detail that internal organs are visible. But for now, this is a living ecosystem, vibrant with activity. As the sun sets over this ancient reef, bioluminescent organisms begin their nightly display. Flashes of blue and green punctuate the darkness. A comb jelly pulses with ribbons of light. A crustacean leaves a trail of glowing particles as it swims. Night brings out different predators. Anomalocaris, which hunted by sight during the day, now relies more on its other senses. It can detect vibrations in the water, chemical traces of prey, perhaps even electrical fields generated by muscle contractions. The darkness doesn't stop the hunt. It merely changes the rules. I spend weeks at this Burgess Reef, trying to catalogue the diversity, to understand the ecological relationships, to appreciate the full symphony of life in this special place. Each day brings new discoveries. A creature I thought was a predator turns out to be a filter feeder. An apparent scavenger is actually a sophisticated hunter. Two species I thought were unrelated proved to be different life stages of the same organism. The Burgess fauna represents the Cambrian explosion at its peak, not just in terms of diversity, but in terms of experimentation. Life is trying everything, exploring every possible way to build a body, to make a living, to interact with other organisms. Most of these experiments will fail in the long term, but some will succeed spectacularly, establishing patterns that persist to this day. As I prepare to leave this remarkable place, I reflect on what makes it so special. It's not just the diversity or the strangeness of the creatures. It's the sense of unlimited possibility, of life unconstrained by tradition or precedent. In later eras, evolution will mostly tinker with established body plans. But here in the Cambrian, everything is new, everything is possible. The Burgess Reef is evolution's symphony hall, where life is performing a composition of unlimited creativity. Some movements will be forgotten, some themes will be abandoned, but the music itself, the endless improvisation of life exploring its possibilities, will continue as long as Earth endures. Before I show you how the patterns of the Cambrian become fixed for all time, let me take you to witness the creation of one of the most magnificent ecosystems ever to exist. The building of the Great Reef that will one day be known as the Burgess Shale. For this is where life's creativity reaches its peak, where the ocean builds a cathedral that will preserve the memory of this golden age forever. I drift back through time to 515 million years ago, to waters that lie along the equator in what your maps would call Western Canada. But there are no continents as you know them, no Rocky Mountains piercing the sky. Instead, I find myself in a vast warm sea, crystal clear and teeming with life. The water temperature hovers around a perfect 80 degrees, heated by a sun that shines without interruption through skies unmarred by ice or storm. At first glance, this seems like any other Cambrian sea, but something special is happening here, something that will create one of the most important fossil sites in Earth's history. Deep currents, rich with nutrients from the world's first upwelling zones, bathe a gently sloping sea floor. The combination of warm water, bright sunlight, and endless food creates perfect conditions for the most ambitious building project life has ever attempted. The builders are sponges, not the simple sponges you might find in a modern bathroom, but giants that grow like underwater redwoods. Some species reach heights of 6 feet or more, their bodies rigid with spicules of glass-like silica. Others spread like living carpets across the sea floor, their surfaces riddled with channels and cavities where water flows in endless circulation. Still others grow in fantastic branching patterns, creating underwater forests of living architecture. But these sponges are not building alone. Partnerships that will define marine life for hundreds of millions of years are forming before my eyes. Tiny organisms, bacteria, and simple algae take up residence within the sponge tissues. The sponges provide a protected home with a steady flow of fresh water. In return, their microscopic partners produce food through the ancient art of capturing sunlight and transforming it into nourishment. I watch as the first small patch of reef begins to grow. A pioneer sponge, no larger than a dinner plate, anchors itself to a stable piece of sea floor. Its porous body acts like a living pump, drawing water through thousands of tiny openings, filtering out bacteria and organic particles for food. The waste products it produces, calcium and other minerals concentrated from the seawater, begin to cement the sediment around its base. This simple beginning attracts others. Young sponges of different species settle on and around the pioneer, each adding their own architectural style to the growing structure. Some grow upward like towers, others spread sideways like living platforms. The spaces between them become channels and caverns where water can flow freely, bringing fresh nutrients and carrying away waste. As the reef grows taller, it creates its own weather system in the water. The massive sponge colonies redirect currents, creating areas of fast-flowing water perfect for filter feeders and quiet pools, ideal for delicate organisms. Turbulence at the reef's edge stirs nutrients up from deeper water, while the sheltered areas behind the reef become nurseries where young creatures can grow protected from strong currents and large predators. The diversity that accumulates around this growing reef is staggering. I catalogue creatures that exist nowhere else on Earth, species that evolution is trying out for the first and only time. Here is a worm with 5 heads arranged in a circle, each head equipped with its own set of eyes and feeding tentacles. There is a creature that looks like a living flower, but captures prey with lightning-fast reflexes. Another appears to be part plant, part animal, and part something else entirely. What makes this reef so special is not just its size. Though it grows to massive proportions, stretching for miles along the seafloor and rising nearly 300 feet above the surrounding plains, but its three-dimensional complexity. Unlike modern reefs, which are largely built by corals, this ancient reef creates countless microhabitats, each with its own community of specialized creatures. In the deepest caverns, where little light penetrates, I find communities of animals that have given up photosynthesis entirely, living instead on the rain of organic matter that filters down from the sunlit regions above. Strange arthropods with enormously enlarged eyes hunt in the perpetual twilight. Soft-bodied creatures that would be quickly eaten in the open ocean find safety in the narrow crevices. Higher up, where light still penetrates but currents are gentle, gardens of delicate organisms flourish. Here I find some of the most beautiful creatures the ocean has ever produced. Animals with bodies like living stained glass, transparent tissues shot through with brilliant colors. They feed by extending gossamer-thin feeding nets that capture microscopic prey with a precision that seems almost magical. At the very top of the reef, where sunlight is brightest and currents strongest, the most active communities thrive. This is the domain of the hunters. Arthropods, with powerful grasping arms, patrol the reef surface, while schools of early swimmers dart between the sponge towers like fish among coral heads. The competition here is fierce, but so is the abundance. Evolution has created a perfect balance between predator and prey. But the reef's greatest innovation is how it handles the challenge of waste. In most ecosystems, waste products accumulate and eventually poison the system. But this reef has evolved a perfect recycling program. Nothing is wasted. The waste products of one organism become food for another. Dead organic matter is quickly broken down and redistributed. Even the calcium carbonate shells of dead creatures are dissolved and reused by the living reef builders. As I observe this magnificent ecosystem over thousands of years, I witness how it shapes its own evolution. The complex three-dimensional structure of the reef creates countless new ways of making a living, creatures evolved to be specialists in navigating narrow channels, feeding in fast currents, or hiding in dark crevices. Each new adaptation opens up more possibilities, driving an accelerating spiral of diversification. The reef also acts as an evolutionary laboratory, bringing together creatures from distant parts of the ocean. When sea levels change or currents shift, refugees from other regions wash up on the reef. Most don't survive, but some find empty niches and establish new populations. This constant influx of genetic diversity provides raw material for evolution to work with. I'm particularly fascinated by how the reef develops its own communication systems. Chemical signals pass through the water like messages, warning of approaching predators or announcing the discovery of food. Some creatures learn to eavesdrop on these chemical conversations, gaining information about distant events. Others evolve the ability to produce false signals, luring prey or confusing competitors. The visual displays that emerge are equally remarkable. In the clear, well-lit waters around the reef, appearance matters as never before. Creatures evolve brilliant colors and intricate patterns, some to attract mates, others to warn of toxicity, still others to blend invisibly with their surroundings. The reef becomes a living art gallery where evolution experiments with every possible combination of color, pattern, and form. But perhaps the most important innovation I observe is the development of ecological partnerships that go far beyond simple predator-prey relationships. Creatures learn to cooperate in ways that benefit both partners. Small arthropods become living cleaners, removing parasites from larger creatures in exchange for a steady food supply. Filter feeders position themselves to catch the waste products of their neighbors, while those neighbors benefit from having their waste removed before it can attract scavengers. Some partnerships become so intimate that the boundaries between species begin to blur. I observe cases where two different types of organisms live in such close association that they can no longer survive independently. Their life cycles become synchronized, their body chemistry integrated, their genetic destinies intertwined. These are the first true symbioses. Partnerships that will become one of evolution's most powerful strategies. As the reef reaches its mature phase, covering hundreds of square miles of sea floor, it faces new challenges. Its very success threatens its survival. The massive structure begins to alter ocean currents on a regional scale, redirecting nutrients away from some areas and concentrating them in others. The reef communities must constantly adapt to these self-imposed changes. Competition within the reef intensifies as space becomes limited. Established communities resist invasion by newcomers. Territorial battles break out between different species of sponges, each trying to claim the best positions for filter feeding. Some creatures evolve chemical weapons to defend their turf, while others develop armor to resist chemical attacks. The arms race that began with simple predation now extends to every aspect of reef life. But the reef's ultimate challenge comes from its own geology. The massive structure, built on a foundation of sediment and shells, becomes unstable as it grows taller. Small collapses occur regularly as sections of the reef outgrow their foundations. These mini-disasters eliminate some species while creating opportunities for others. The reef learns to live with constant renewal, a cycle of destruction and rebuilding that keeps evolution in constant motion. The final chapter in the reef story begins 510 million years ago, when the geological stability of the region starts to change. Tectonic forces deep beneath the ocean floor begin to tilt the sea floor, creating an unstable slope along the reef's edge. Earthquakes become more frequent, shaking loose vast sections of the reef structure. At first, These collapses are minor disasters, destroying local communities, but allowing the reef as a whole to continue growing. But gradually, the earthquakes become more severe. Massive underwater landslides begin to sweep down the tilted sea floor, carrying everything in their path: living animals, empty shells, reef debris, even chunks of living reef, into the deep basins beyond. I position myself to witness one of these catastrophic slides. The seafloor trembles, then gives way entirely. A wall of mud and debris 300 feet high cascades over the reef edge like a liquid avalanche. Everything in its path is instantly buried. Animals caught in the middle of feeding, others fleeing frantically, but unable to escape the advancing wall of sediment. The destruction is complete within minutes, but the burial conditions are perfect for preservation. The fine mud excludes oxygen, preventing decay. The rapid burial means that even the most delicate soft tissues are protected from scavengers. What begins as a disaster ends as a perfect preservation event, creating fossil deposits of extraordinary detail. This process repeats hundreds of times over several million years. Each major earthquake triggers new avalanches. Each avalanche buries a snapshot of reef life in perfect detail. Layer upon layer of these burial events accumulates in the deep basin, creating what will eventually become the Burgess Shale, one of the most important fossil deposits in the history of science. But I'm witnessing more than just the formation of a fossil site. I'm observing the end of an era. The reef ecosystem that supported such extraordinary diversity cannot survive the geological upheaval. The earthquakes fragment the reef into isolated patches, breaking up the ecological connections that sustain the complex communities. Many of the most specialized species, adapted to very specific reef habitats, cannot survive in the simplified environments that replace the reef. Yet even in destruction, the reef leaves a lasting legacy. The species that survive the collapse carry the genetic innovations developed in the reef to other parts of the ocean. The biochemical pathways for building shells, the neural circuits for processing complex visual information, the behavioral programs for cooperative interactions. All these innovations spread throughout the Cambrian seas. More importantly, the reef has proven that life can build ecosystems of almost unlimited complexity, given the right conditions. The architectural principles it developed, the use of three-dimensional structure to create multiple habitats, the integration of recycling systems, the evolution of cooperative partnerships, will be rediscovered again and again throughout Earth's history. As the last great avalanche buries the final remnants of the reef, I'm struck by the irony that this destruction ensures the reef's immortality. The very slides that ended the living ecosystem created the conditions that preserved its memory in perfect detail. 500 million years in the future, scientists will split open rocks formed from these buried sediments and marvel at the creatures revealed within. They will see Opabinia with its 5 eyes, an elephant-like trunk, Hallucigenia walking on its spines, Wiwaxia armored like a medieval knight. They will debate the relationships of these ancient creatures, using them to understand how modern animal groups evolved. They will recognize in these fossils the experimental phase of evolution, when life was trying every possible solution to the challenges of multicellular existence. But what those future scientists cannot fully capture is what I have witnessed: the living, breathing ecosystem in all its complex glory. They will see the individual creatures but miss the elaborate partnerships that sustain them. They will marvel at the bizarre forms but not understand the ecological pressures that shape them. They will count the species but cannot measure the symphony of interactions that made the reef a living community. The Burgess Reef represents the peak of Cambrian creativity, the moment when life's experimentation with form and function reached its greatest intensity. It shows what becomes possible when evolution has both the raw materials and the opportunity to innovate freely. But it also demonstrates the fragility of complexity, how quickly elaborate ecosystems can be destroyed by changes in their physical environment. As I prepare to leave this buried cathedral, I carry with me a profound appreciation for the creative power of evolution. In building the Burgess Reef, life showed that it could construct ecosystems as complex and beautiful as anything your civilization has ever built. The reef was evolution's masterpiece, a demonstration of what becomes possible when countless small innovations combine to create something greater than the sum of its parts. The reef is gone now, its living communities scattered to the winds of geological time. But its fossils remain, buried in the mountains of Western Canada, waiting to tell their story to anyone wise enough to listen. In those ancient stones lies the memory of evolution's greatest experiment, preserved forever in the tomb of time. As the Cambrian period progresses through its 56 million year span, I observe the initial explosion of diversity beginning to channel into patterns. The wild experimentation of the early Cambrian gives way to refinement and specialization. Body plans that prove successful diversify into multiple species. Those that don't gradually disappear. I position myself in the Middle Cambrian seas, about 510 million years ago, to observe how the innovations of the explosion are being consolidated. The major animal groups, what your scientists call phyla, have been established. Now evolution is exploring variations within these basic blueprints. The arthropods have emerged as the clear winners in terms of diversity and abundance. Their modular body plan, repeating segments that can be modified for different functions, proves incredibly versatile. I observe hundreds of arthropod species, each with its own variation on the theme. The trilobites, in particular, undergo spectacular diversification. Where once there were a handful of species, now there are hundreds. Some develop elaborate spines for defense. Others evolve shovel-shaped heads for plowing through sediment. Some become swimmers, their bodies streamlined and their eyes enormous. Others become burrowers, their eyes reduced to tiny spots or lost entirely. I follow the life cycle of one trilobite species to understand their success. The female releases hundreds of tiny eggs into the water. Each egg hatches into a larva, less than a millimeter long, with only a head shield and no body segments. As it grows and molts, it adds segments one at a time, gradually developing the three-lobed body form of an adult. This growth by molting is key to arthropod success. Unlike creatures that grow continuously, arthropods can radically restructure their bodies at each molt. A larva specialized for dispersal can transform into a juvenile specialized for hiding, then into an adult specialized for reproduction. It's like having multiple body plans in a single lifetime. But arthropods aren't the only success story. The mollusks are diversifying into forms that will become familiar in later seas. I observe early gastropods, snails, with their characteristic coiled shells. The coiling is more than aesthetic. It allows the shell to grow indefinitely while maintaining structural strength and balance. Early cephalopods are experimenting with buoyancy control. They build shells divided into chambers, filling some with gas to achieve neutral buoyancy. A primitive nautiloid demonstrates this innovation as it adjusts its depth, pumping fluid in and out of its chambers to rise or sink. It's a biological submarine, able to move vertically through the water column without swimming. The brachiopods, those lamp shells that superficially resemble clams, reach their peak diversity in these Cambrian seas. Unlike mollusks, which are related to snails and squids, brachiopods are their own phylum with a unique body plan. I watch them filter feeding, their shells opening and closing in rhythm, internal cilia creating currents that bring in food particles. Each major group has found its niche. Echinoderms, the ancestors of starfish and sea urchins, developed their characteristic five-fold symmetry and water vascular system. Primitive chordates, our own distant ancestors, refine their body plan of notochord, dorsal nerve cord, and segmented muscles. Cnidarians, jellyfish and their relatives, perfect their simple but effective combination of stinging cells and radial symmetry. But what's most remarkable is not the diversity of forms, but the establishment of ecological relationships that will persist for hundreds of millions of years. The basic structure of marine ecosystems: primary producers, herbivores, primary carnivores, apex predators, decomposers, is fully established. I observe a food web that could almost be modern in its complexity. Photosynthetic plankton and bacterial mats form the base. These are grazed by numerous small arthropods, mollusks, and worms. Small predators hunt the grazers. Larger predators hunt the hunters. When any organism dies, bacteria and scavengers recycle its nutrients back into the system. The establishment of this ecological structure drives further evolution. Once roles are defined, organisms specialize to fill them more efficiently. Herbivores evolve better grinding mouth parts and longer guts to extract more nutrition from plant material. Predators evolve faster swimming, better senses, more effective weapons. An evolutionary arms race develops that will continue indefinitely. But before we explore how these patterns become fixed, let me take you deep into the molecular world to witness one of the most important innovations of the Cambrian explosion. The moment when life learned to build with stone. To truly understand the transformation of the Cambrian seas, I must show you something almost invisible, yet revolutionary. The moment when life first learned to turn seawater into solid armor. This is the story of biomineralization, one of the most important innovations in the history of life, told at the scale of molecules and cells. I shrink my perspective down to the microscopic level, following the life of a pioneer creature that your scientists will call Claudina. She is tiny, no longer than a grain of rice, and to most observers, utterly unremarkable. But within her cells, a revolution is beginning that will transform every ocean on Earth. The seawater around Claudina is rich with dissolved minerals, calcium ions drifting like invisible snow through the crystal clear water. These calcium particles come from the weathering of rocks on distant landmasses, carried to the sea by rivers and streams. For billions of years, these minerals have simply accumulated in the oceans, with no living creature able to use them for construction. But Claudina's ancestors have evolved something unprecedented. Special cells that can capture these dissolved minerals and transform them into solid structures. I position myself within one of these cells to witness this molecular miracle firsthand. The cell is a bustling factory of biological activity. Protein machines work around the clock, powered by the energy of life itself. Some proteins act as pumps, drawing calcium ions from the surrounding seawater and concentrating them within the cell. Others produce carbonate ions through the breakdown of carbon dioxide. Still others create the organic framework that will guide the crystal formation. The process begins with a decision, which sounds strange when talking about a single cell, but this primitive organism has evolved the ability to sense when shell building is needed, when the cell detects threats in the environment. Perhaps the chemical signatures of predators, perhaps physical damage to existing shell. It activates its construction program. First, the cell creates a special space where the shell will form. This isn't just an empty cavity, but a carefully controlled environment, isolated from the chaotic chemistry of the ocean. The cell lines this space with a thin membrane studded with specialized proteins. These proteins act like molecular architects, determining where and how the shell material will be deposited. The actual construction is a marvel of precision engineering. The cell releases calcium ions into this controlled space, but not randomly. The organic framework proteins arrange themselves in precise patterns, creating templates that guide the crystal formation. It's like building a house with a blueprint that's made of the same material as the house itself. As the calcium and carbonate ions meet in this organized environment, they begin to form crystals of calcium carbonate. But these aren't the chaotic crystals you might find in a cave. The organic proteins control every aspect of the crystal growth: their size, their orientation, their internal structure. The result is a material that combines the hardness of stone with the precision of biological engineering. The first shells are simple, just tiny caps of mineral that can protect the soft parts of the organism. But even this simple innovation provides an enormous advantage. When a predator attacks, Claudina can withdraw into her mineral fortress, protected by walls harder than anything the predator has encountered before. But building with minerals requires solving problems that no organism has faced before. Calcium carbonate is heavy, much heavier than the organic tissues it protects. Too much shell, and the organism becomes too heavy to move effectively. Too little, and the protection is inadequate. Evolution must find the perfect balance. The solution emerges through millions of tiny experiments. Some individuals produce shells that are too thick, making them sluggish and vulnerable to faster predators. Others produce shells that are too thin, offering inadequate protection. But a few hit the optimal balance. Just enough mineral to provide protection without sacrificing mobility. As I observe this process over thousands of generations, I witness the refinement of biological architecture. The simple cone shape of early shells gives way to more sophisticated designs. Some species evolve shells with internal chambers that provide strength without excess weight. Others develop shells with intricate patterns of ridges and grooves that deflect attacks while using minimal material. The chemistry of shell building also evolves rapidly. Early organisms can only work with calcium carbonate, but soon some species learn to incorporate other minerals. Phosphate-based shells appear, offering different properties, lighter but more flexible than carbonate shells. Silica-based structures emerge, creating shells as clear as glass, but harder than granite. Each mineral requires its own construction techniques. Calcium carbonate can be grown slowly, layer by layer, like building with bricks. But silica must be deposited quickly from a concentrated solution, like casting metal in a mold. Phosphate falls somewhere in between, requiring careful control of pH and temperature. Life learns to be a master chemist, working with whatever raw materials are available. The most remarkable innovation is the development of composite materials, shells that combine organic and mineral components in ways that exceed the properties of either material alone. I observe cells that create shells with alternating layers of mineral and protein, like microscopic plywood. The result is a material that's both strong and flexible, able to resist both crushing and piercing attacks. Other species develop shells with different properties in different regions, The outer surface might be hard and smooth to resist abrasion, while the inner surface is textured to provide attachment points for muscles. Some areas might be thick for maximum protection, while others are thin to allow for growth or flexibility. As shell-building techniques spread throughout the Cambrian seas, an architectural arms race begins. Predators evolve stronger jaws and drilling capabilities to penetrate shells. Prey respond by developing thicker shells, more complex shapes, and chemical defenses. Each innovation in defense drives innovation in offense, and vice versa. I'm particularly fascinated by how organisms solve the problem of growth. Unlike the exoskeletons of arthropods, which must be shed and replaced periodically, shells can grow continuously. But this requires extraordinary coordination between the living tissue and the mineral structure. The solution is to build shells in zones. The organism adds new material only at specific growth edges, usually around the opening of the shell. Special cells at these edges work continuously, laying down new layers of mineral as the organism grows. The process is so precise that growth patterns are preserved in the shell structure like tree rings, creating a permanent record of the organism's life history. Some species take this growth pattern to extraordinary extremes. Spiral shells appear, growing in logarithmic patterns that maintain optimal shape as the organism increases in size. These mathematical curves, discovered by human mathematicians millions of years later, emerge naturally from the constraints of biological growth and mineral deposition. The ecological effects of widespread shell building are profound. For the first time, dead organisms leave substantial remains. Shell beds begin to accumulate on the seafloor, creating new habitats and changing the chemistry of marine sediments. The calcium cycle, previously controlled entirely by physical processes, now has a biological component. These shell accumulations become the foundation for new ecosystems. Small organisms find shelter in empty shells, encrusting species attached to shell surfaces. Burrowing organisms mine shells for calcium, recycling the mineral through biological pathways. What begins as protection for individual organisms becomes the building material for entire communities. But perhaps the most important consequence of shell building is what it does to the fossil record. For 3 billion years, life left few traces of its existence. Soft tissues decay quickly, leaving only the occasional bacterial mat or chemical signature. But shells preserve, creating the first detailed record of animal evolution. This preservation revolution means that evolution itself begins to change. For the first time, innovations in shell design can be preserved and passed down through generations, not just genetically, but physically. Organisms can learn from the preserved shells of their ancestors, using them as templates for their own construction projects. The variety of shell designs that emerges is staggering. Some organisms build shells that are works of art, intricate spirals decorated with ribs, spines, and patterns of extraordinary beauty. Others focus purely on function, creating shells optimized for specific environments or lifestyles. Still others experiment with entirely novel approaches, combining shells with other structures in ways never tried before. I observed the development of modular shell systems, armor made of multiple pieces that can move independently. This allows organisms to have protection while retaining flexibility. Trilobites perfect this approach, creating exoskeletons composed of hundreds of individual pieces that work together like medieval chainmail. Bivalved shells appear, hinged structures that can open for feeding and close for protection. This innovation proves so successful that it will be reinvented multiple times by different groups. Clams, oysters, and brachiopods all arrive at similar solutions to the same engineering challenges. Some of the most creative solutions emerge from the interaction between shell building and other biological processes. Organisms that use shells for protection also discover they can use them for buoyancy control. By adjusting the gas content of internal chambers, they can rise or sink in the water column at will. This innovation leads to some of the most sophisticated predators of the Cambrian seas. The chemistry of shell formation also drives innovations in other areas of biology. The proteins used to control crystal growth turn out to have applications beyond shell building. Some are adapted for creating adhesives that allow organisms to stick to surfaces. Others are modified to produce toxins or digestive enzymes. The biochemical toolkit developed for mineralization becomes a resource for solving many different biological problems. As I observe these developments over millions of years, I'm struck by how biomineralization becomes both an enabling technology and a constraint. Organisms that learn to build with minerals gain access to new ways of life. They can be larger, more heavily defended, more structurally complex, but they also become dependent on specific ocean chemistries and environmental conditions. Changes in seawater chemistry that would have no effect on soft-bodied organisms can devastate shell builders. If calcium levels drop or pH changes, entire ecosystems of mineralized organisms can collapse. The power of biomineralization comes with a price. Dependence on the geochemical conditions that make it possible. Yet for all its risks, biomineralization proves to be one of evolution's most successful innovations. Once established in the Cambrian, it never disappears. Every major group of marine organisms eventually develops some form of mineral construction. Vertebrates build skeletons of calcium phosphate. Arthropods create exoskeletons of calcified chitin. Mollusks perfect shells of incredible diversity and beauty. The techniques pioneered by those first Cambrian shell builders are still in use today. The proteins that control crystal formation in modern shells are remarkably similar to those I observe in these ancient seas. The basic principles of biological architecture, controlled nucleation, organic templating, composite construction, remain unchanged after 500 million years. As I prepare to return to the larger scale of whole organisms and ecosystems, I carry with me a deep appreciation for what I've witnessed at the molecular level. The evolution of biomineralization wasn't just the development of a new technology. It was the moment when life learned to collaborate with geology, to harness the planet's own materials for biological purposes. This collaboration between life and stone will prove to be one of the most durable partnerships in Earth's history. From the first simple shells of Cloudina to the elaborate skeletons of modern animals, the union of organic and mineral materials has provided the foundation for most complex life. In learning to build with stone, life discovered not just protection, but permanence, the ability to create structures that outlast individual organisms and preserve the memory of evolution itself. The shells of the Cambrian seas were more than armor. They were the first libraries, storing information about the history of life in their crystalline structures. And in the mountains, where these shells are now preserved as fossils, that library remains open, waiting to tell its story to anyone wise enough to read the language of stone. I'm particularly intrigued by the evolution of developmental patterns that will constrain all future evolution. The bilateral symmetry established in the Cambrian, with distinct front and back ends, left and right sides, becomes almost universal among mobile animals. Radial symmetry is relegated to sessile forms like sea anemones or slow-moving forms like sea stars. The segmentation pioneered by arthropods and annelid worms proves so useful that it appears independently in multiple groups. Even vertebrates show segmentation in their muscle blocks and vertebral columns. It's a pattern that allows for both flexibility and specialization, with different segments adapted for different functions. The concentration of sensory organs and neural tissue at the front end, cephalization, becomes standard for mobile animals. It makes sense. The end that enters new environments first should be the one that senses and processes information. This pattern, established in the Cambrian, will constrain body design forever after. I observe how these developmental constraints channel evolution. A mutation that would produce a 5-legged arthropod is theoretically possible, but it would disrupt the bilateral symmetry that governs development. Such mutations occur but don't persist. The body plans established in the Cambrian become canalized, difficult to fundamentally alter. Yet within these constraints, the variations are endless. I catalogue trilobites with every possible arrangement of spines, every possible eye configuration, every possible body proportion. They're all variations on the trilobite theme, but each species has found its own unique solution to survival. The late Cambrian seas, around 490 million years ago, show both the triumph and the limitations of the explosion. Diversity at the species level continues to increase, but the fundamental body plans are set. No new phyla will appear. Evolution will work with what the Cambrian has provided, refining and elaborating, but not fundamentally innovating. I witness several minor extinction events that punctuate the late Cambrian. Changes in sea level, ocean chemistry, or climate periodically stress the ecosystem. Each time, some species disappear, while others expand to fill the empty niches. But the basic patterns persist. Arthropods remain arthropods. Mollusks remain mollusks. Chordates remain chordates. The very success of the Cambrian explosion may have limited future possibilities. With all major niches filled, with successful body plans established, there's less room for fundamental innovation. A new body plan would have to compete with well-adapted existing forms. The window of opportunity that the Cambrian represented is closing. Yet the legacy is profound. In establishing the major animal phyla, the Cambrian set the stage for all future evolution. The arthropod body plan will be modified to produce insects, spiders, and crustaceans. The chordate body plan will give rise to fish, amphibians, reptiles, birds, and mammals. The patterns established here will constrain and channel evolution for the next 500 million years. As I prepare to leave the Cambrian seas, I reflect on what I've witnessed. In perhaps 20 million years, a geological instant, life transformed from simple, soft-bodied forms to complex ecosystems filled with shells, eyes, and elaborate behaviors. It was the most concentrated period of evolutionary innovation in Earth's history. The Cambrian explosion was many things. It was an arms race between predator and prey, driving the evolution of weapons and defenses. It was an information revolution, as nervous systems became complex enough to process sensory data and coordinate responses. It was an architectural breakthrough, as life learned to build with minerals. But perhaps most fundamentally, it was the moment when life established the patterns that would govern its future. The body plans, the developmental programs, the ecological relationships formed in the Cambrian would prove remarkably durable. Mass extinctions would come and go, eliminating species and even orders and classes. But the phyla established in the Cambrian would persist. When you look at any animal today, a butterfly, a spider, a fish, a bird, You are looking at the legacy of the Cambrian explosion. The fundamental body plan was established then. The sensory systems were invented then. The ecological role was pioneered then. Everything since has been variation on themes established 500 million years ago. The Cambrian explosion shows both the power and the constraints of evolution. Given the right conditions, rising oxygen, available minerals, ecological opportunity, evolution can produce tremendous innovation in a short time. But once successful patterns are established, they become difficult to change. Evolution is both creative and conservative, capable of explosive change and remarkable stability. This is the lesson of the Cambrian. Life is endlessly inventive within the patterns it establishes. The explosion produced the themes. Everything since has been variations. And what wonderful variations they have been. From trilobites to butterflies, from Pikaia to humans, from simple compound eyes to the visual systems that let you read these words. The Cambrian explosion was life's Big Bang, the moment when animal life as we know it came into being. Its echoes reverberate through every ecosystem on Earth today. Before I return you to your own time, let me show you something profound and personal. As you lie there listening to this story, your body carries within it the living memory of the Cambrian explosion. Every breath you take, every heartbeat, every thought passing through your mind connects you directly to those ancient seas. You are not just a descendant of the Cambrian. You are the Cambrian, transformed but never forgotten. Let me start with your eyes, the organs through which you perceive light and form and color. Close them now, if you haven't already, and imagine the evolutionary journey they've taken to reach you. The fundamental design, a transparent window focusing light onto a layer of sensing cells, was perfected in the Cambrian seas by creatures like the early mollusks I observed. The proteins that detect light in your eyes are nearly identical to those that first appeared in trilobite compound eyes 500 million years ago. Rhodopsin, the molecule that transforms photons into electrical signals, uses the same basic mechanism in your eyes as it did in those ancient arthropods. The genetic instructions for building eyes, the master control gene called PAC6, is so fundamental that it controls eye development in creatures as different as insects and humans. When you see a sunset, when you read these words, when you recognize the face of someone you love, you're using biological machinery that was invented during the Cambrian explosion. Your visual system is a direct descendant of those first experiments with sight that transformed the ancient ocean from a world of darkness into a realm of light and shadow. But your connection to the Cambrian runs much deeper than just your eyes. Consider your skeleton, the internal framework that supports your body and protects your vital organs. This innovation, too, has its roots in the Cambrian seas. The earliest vertebrates, humble creatures like Pikaia, pioneered the concept of an internal supporting rod, the notochord, that would eventually evolve into your backbone. Every bone in your body is built using the same basic biochemical processes that Cambrian organisms developed for shell construction. The proteins that control crystal formation in your bones are modified versions of the same proteins that built the first shells. Your skeleton is essentially an internal shell, using the same mineral, calcium phosphate, that some Cambrian creatures used to build their protective armor. When you stand upright, when you walk, when you run, you're using a skeletal system that represents 500 million years of refinement of those first Cambrian experiments with biomineralization. Your bones are living testimonies to the moment when life first learned to build with stone. Your heart That tireless muscle that has been beating since before you were born also carries the memory of Cambrian innovation. The basic plan for a circulatory system, a muscular pump pushing fluid through a network of tubes, was established in those ancient seas. The genes that control heart development, the proteins that make heart muscle contract, the very architecture of your cardiovascular system, all have their origins in the body plans that emerged during the Cambrian explosion. Every time your heart beats, it's performing a function that was perfected by the first complex animals. The four-chambered heart of mammals is an elaboration of the simple pumping systems that appeared in early arthropods and mollusks. Your circulation carries the same basic elements: oxygen, nutrients, waste products that were transported through the bodies of trilobites and early vertebrates. Even more fundamental is your nervous system, the network of electrical communication that allows you to think, feel, and respond to the world around you. The basic architecture of nerve cells, the way they generate and transmit electrical signals, the chemical messengers they use to communicate, all of these were established during the Cambrian explosion. Your brain, for all its sophistication, is built on the same basic plan as the simple nerve clusters that appeared in early arthropods. The concept of centralized information processing, of sensory organs sending signals to a central decision-making structure, was pioneered by creatures swimming in the Cambrian seas. When you think, when you remember, when you dream, you're using neural networks that trace their ancestry back to those first experiments with complex behavior. The neurotransmitters that carry signals between your nerve cells, serotonin, dopamine, acetylcholine, are ancient molecules that appeared in the Cambrian and have been conserved ever since. The feelings of pleasure, fear, curiosity, and wonder that you experience are generated by chemical systems that first evolved to help early animals navigate their world. Your digestive system, too, carries the Cambrian blueprint. The basic plan, a tube passing through your body with specialized regions for different aspects of food processing, was established by the first bilaterally symmetric animals. Your mouth, stomach, liver, and intestines are all elaborations of the simple gut systems that appeared during the explosion. The enzymes that break down your food, the mechanisms that absorb nutrients, the systems that eliminate waste, all have their origins in those early experiments with complex, multicellular life. When you eat, you're using digestive processes that were first developed by creatures feeding on Cambrian algal mats. Even your immune system, that sophisticated defense network that protects you from disease, has Cambrian roots. The basic concept of distinguishing self from non-self, of identifying and eliminating threats to the body's integrity, emerged with the first complex animals. The proteins that recognize foreign invaders, the cells that mount immune responses, the memory systems that allow you to resist reinfection, all are descendants of the defensive mechanisms that evolved during the Cambrian arms race between predators and prey. Your reproductive system carries perhaps the most ancient memories of all. The basic processes of sexual reproduction, of combining genetic material from two individuals to create offspring, predate the Cambrian. But the complex hormonal systems that control reproduction, the elaborate behaviors associated with finding and choosing mates, the developmental programs that build new organisms from single cells, all were refined and perfected during the Cambrian explosion. When you experience attraction, when you form pair bonds, when you care for offspring, you're expressing behavioral and physiological programs that were shaped by the evolutionary dynamics of those ancient seas. The hormones that influence your social behavior, testosterone, estrogen, oxytocin, are molecular messengers that first appeared in early vertebrates and arthropods. But perhaps the most profound connection between you and the Cambrian lies in your development from a single fertilized egg into a complex multicellular organism. The genetic program that controls this transformation, the master control genes, called Hox genes, was established during the Cambrian explosion and remains virtually unchanged in all animals today. The same genes that told a trilobite's body which end should be the head and which should be the tail control the development of your own body axis. The genes that determined how many segments a Cambrian arthropod would have influenced the number of vertebrae in your spine. The genetic cascades that built the first compound eyes guide the formation of your own visual system. When you were developing in the womb, your body briefly recapitulated key stages of Cambrian evolution. You had gill slits like an early vertebrate, a tail like Picaia, segments like an arthropod. Your embryonic development is a compressed replay of the evolutionary innovations that occurred during the explosion, a biological history lesson written in your own flesh. This deep connection to the Cambrian means that you carry within yourself the memory of every major evolutionary innovation that occurred during the explosion. Your bilateral symmetry remembers the first animals that developed distinct head and tail ends. Your complex sensory organs remember the moment when life discovered the power of perception. Your elaborate body systems remember the transition from simple to complex, from single-celled to multicellular, from basic survival to sophisticated behavior. You are not just related to the creatures of the Cambrian seas. You are those creatures, transformed by 500 million years of evolution, but still fundamentally the same. The innovations that seem so strange and experimental in those ancient waters have become the foundation of your own existence. Every cell in your body operates according to principles established during the explosion. This realization should fill you with wonder. You are the living embodiment of the Cambrian explosion's greatest achievements. The creativity, the experimentation, the endless innovation that characterized those ancient seas continues in you. Every adaptation you make, every challenge you overcome, every new skill you learn is a continuation of the evolutionary process that began in the Cambrian. When you use your eyes to appreciate beauty, you're exercising the gift of sight that was born in the Cambrian. When you use your brain to solve problems, you're employing the intelligence that first emerged in those ancient arthropods. When you form relationships with others, you're expressing the social behaviors that evolved in the complex ecosystems of the explosion. You are not separate from nature. You are nature, conscious of itself. You are not apart from evolution. You are evolution, aware of its own history. The Cambrian explosion didn't just create the body plans that would dominate life on Earth. It created the capacity for consciousness, for self-awareness, for the ability to contemplate one's own existence. In you, the Cambrian explosion achieved something that would have been impossible in those ancient seas. The ability to look back through time and understand its own significance. You are the Cambrian's gift to the universe, a way for the cosmos to know itself, to appreciate its own creativity, to marvel at the unlikely journey from simple chemicals to conscious beings capable of love, art, science, and wonder. As you drift towards sleep, carrying this story with you, remember that you are not just listening to ancient history. You are ancient history, come alive and aware. Every dream you have tonight will be dreamed by a mind built on Cambrian foundations. Every breath you take will be taken by lungs derived from Cambrian innovations. Every heartbeat will pulse through a circulatory system perfected in those distant seas. You are the Cambrian explosion made conscious, the flower of evolution in full bloom, the universe awakening to its own magnificence. In you, those ancient seas live on, not as memory, but as reality, not as past, but as present. The explosion that began 500 million years ago continues in your every thought, your every action, your every moment of being alive. This is your deepest connection to the story I've shared, not as an observer, but as a participant, not as a descendant, but as a continuation. You are the Cambrian explosion, still exploding, still creating, still becoming something new and wonderful with each passing day. As I prepare to return you to your own time, I want to share how the innovations of the Cambrian explosion echo through all subsequent history, shaping life on Earth to this very day. I drift once more above these ancient seas, now at the very end of the Cambrian period, 485 million years ago. The explosion is over, but its reverberations are just beginning. The major animal groups are established. The basic ecological relationships are in place. The arms race between predator and prey is in full swing. This is no longer an experimental ecosystem, but a mature one, with patterns that will persist through unimaginable changes to come. Looking forward through time from this vantage point, I can see how the Cambrian innovations will play out. The compound eyes of trilobites will be refined and perfected in insects and crustaceans. Some dragonflies will develop vision so acute they can detect the movement of individual wing beats from across a pond. Mantis shrimps will evolve eyes with 16 types of color receptors compared to your 3, seeing colors you cannot imagine. The simple camera eyes of mollusks will undergo their own spectacular evolution. Octopuses and squids will develop eyes as sophisticated as those of vertebrates, with focusing lenses, adjustable pupils, and high-resolution retinas. It's a case of convergent evolution. Two lineages independently arriving at similar solutions to the challenge of forming sharp images underwater. The shells that first appeared in the early Cambrian will be elaborated into countless forms. Mollusks will produce shells of extraordinary beauty and mathematical precision. The logarithmic spirals of nautiluses, the complex patterns of cone shells, the iridescent nacre of abalone. Each shell will be a work of art, but also a functional structure optimized for protection, buoyancy, or burrowing. The arthropod body plan, so successful in the Cambrian seas, will prove even more successful on land. When plants colonize the continents, arthropods will follow. The modular construction that allowed trilobites to diversify will allow insects to become the most diverse group of animals on Earth. Wings will be added to the basic plan, opening up the aerial realm. Social behaviors will evolve, leading to the complex societies of ants, bees, and termites. But perhaps nowhere is the Cambrian legacy more profound than in the chordates. That simple body plan, notochord, dorsal nerve cord, segmented muscles, post-anal tail, will prove capable of extraordinary modification. The notochord will be replaced by a jointed vertebral column. The nerve cord will expand into a complex brain and spinal cord. The segmented muscles will power everything from the sinuous swimming of eels to the powered flight of birds to the bipedal walking of humans. I think of Pikaia, that humble 2-inch swimmer in the Cambrian seas, and marvel at its descendants. Fish will diversify into tens of thousands of species, from tiny gobies to massive whale sharks. Some will develop lungs and venture onto land, becoming amphibians. Some amphibians will develop watertight eggs and become reptiles. Some reptiles will develop warm blood and become mammals and birds. Each innovation builds on the Cambrian foundation. The basic vertebrate eye, already present in primitive form in Cambrian chordates, will be enhanced with focusing lenses, color-detecting cones, and image-processing retinas. But the fundamental structure, a camera-type eye with a lens focusing light on photoreceptors, remains unchanged from its Cambrian origins. The ecological relationships established in the Cambrian will also persist. The food web structure, primary producers, herbivores, multiple levels of carnivores, decomposers, will be replicated in every ecosystem on Earth. Whether in a tropical rainforest, a grassland savanna, or a deep ocean trench, the same basic patterns apply. Energy flows from producers to consumers, nutrients cycle through the system, predators and prey engage in endless evolutionary arms races. The Cambrian explosion also established the pace and pattern of evolution itself. The rapid diversification when new opportunities arise, followed by refinement and specialization, will be repeated after every mass extinction. When the dinosaurs disappear, mammals will explosively diversify to fill empty niches, just as Cambrian animals did. When humans transport species to new continents, they will undergo rapid evolution to adapt to new conditions, following patterns established 500 million years ago. Even the developmental programs that build bodies show their Cambrian origins. The Hox genes that control segmentation in fruit flies are nearly identical to those that control segmentation in humans. The genetic toolkit for building eyes is shared across the animal kingdom. A gene called PAX6 controls eye development in creatures as different as flies and mice, a shared inheritance from Cambrian ancestors. This deep genetic similarity reveals the profound truth of the Cambrian explosion. All animals are variations on themes established then. The last common ancestor of insects and humans lived in the Cambrian seas. The genetic instructions for building complex bodies were written then, in the DNA of creatures we would barely recognize as relatives. As you return to your own time, carry with you an appreciation for your Cambrian heritage. Your eyes, though vastly more sophisticated than those of a trilobite, work on the same principles and derive from the same evolutionary innovation. Your backbone, though supporting an upright posture no Cambrian creature achieved, is a direct modification of the notochord that first appeared then. Even your behaviors have Cambrian roots. The fight-or-flight response that floods your body with adrenaline when threatened is a refined version of escape behaviors that first evolved when predators appeared. The careful assessment of potential mates, the protection of offspring, the formation of social groups, all have their origins in the behavioral innovations of the Cambrian. When you walk along a beach and see shells washed up by the tide, remember that you're looking at an invention of the Cambrian explosion. When you watch a documentary about the complex eyes of jumping spiders or the sophisticated hunting strategies of octopuses, you're seeing elaborations of Cambrian innovations. When you pet a dog or cat, you're interacting with a fellow vertebrate whose body plan was established alongside trilobites and anomalocarids. The Cambrian explosion was not just an event in the distant past. It was the founding moment of the animal kingdom as we know it. Every animal alive today is a survivor of that explosion, carrying forward innovations and patterns established in those ancient seas. We are all children of the Cambrian, heirs to the creative burst that established the possibilities and constraints of animal life. In your time, as humans drive what many scientists call the sixth mass extinction, it's worth remembering the lessons of the Cambrian. Diversity, once lost, takes millions of years to restore. Body plans, once extinct, do not re-evolve. The wonderful forms that vanish in our time. Each species of butterfly, each type of coral, each kind of frog, represent branches of the Cambrian radiation that will never grow again. Yet the Cambrian also teaches resilience. Life, given opportunity, explodes in creativity. New forms arise to fill empty niches. Evolution finds ways around apparent limitations. The same innovative spirit that produced the Cambrian explosion continues to operate, creating new species even as others disappear. Perhaps humans themselves represent a new kind of explosion, not of body forms but of behaviors, technologies, and ideas. The rapid cultural evolution of humanity, the explosion of information and innovation, mirrors in some ways the biological explosion of the Cambrian. Just as that ancient explosion established patterns that would constrain future evolution, the choices made in your time may establish patterns that constrain future human development. The Cambrian explosion reminds us that moments of explosive change are both creative and defining. They open new possibilities while closing others. They establish patterns that persist long after the explosion ends. Understanding this can help us navigate our own explosive moment with wisdom. As you drift back to consciousness in your own bed, in your own time, remember that you carry within you the legacy of the Cambrian seas. Every cell in your body contains DNA sequences that first appeared then. Every protein that builds your tissues uses a genetic code established then. Every sensory experience you have depends on innovations that arose then. You are not separate from this story, but its continuation. The explosion that began in those warm seas 500 million years ago continues in you. The creativity of evolution, the endless experimentation with form and function, the drive to perceive and respond and survive. All of this lives on in you. When you open your eyes, you will do so with organs whose basic design was established in the Cambrian. When you move, you will do so with muscles arranged in patterns first seen in Cambrian swimmers. When you think about what you've heard, you will do so with a brain built on a Cambrian foundation. You are a living fossil of the Cambrian explosion, a walking museum of 500 million year old innovations. But you are also something new, something the Cambrian could never have imagined. A conscious being capable of understanding its own history, of appreciating the deep connections between all life, of choosing how to shape the future. This is the ultimate gift of the Cambrian explosion. Not just eyes to see and brains to think, but the capacity to understand our place in the grand story of life. Use this gift wisely. The explosion continues, and you are part of it. Until we meet again in the deep rivers of time, I am the Chrononaut, your guide through Earth's memories. Sleep well, child of the Cambrian dawn, heir to the explosion that made all complex life possible. Rest in the knowledge that you are part of life's greatest creative moment, a moment that echoes still in every heartbeat, every breath, every dream. Rest.